This is which set? S3 only, no? Hello, good evening, students. I hope so. You have given your best in your exam. Fine. So this is the time to finalize the thing. But we have tried to give the most authentic type of the key. But still, we will be waiting for the key given by from backside from the authentic sources. Then we will finalize all the things, right? So right now we are taking S3 as a code for the discussion of the question paper, right? So that S3 code is there in front of you. It's starting with the bio part. First of all, botany question, the based on things what we have taught you. So accordingly, we are dividing our session today. I'm taking botany for that, and Uma ma'am is taking for the zoology.
Hello.
Right? So we together are starting for the bio. So first I'm taking for botany session as per the question is given on the botany. So the question number 101. See what they are saying here. The lactose present in the growth medium, bacteria is transported to the cell by the action of which enzyme? This is based on lactopron, right? So that the lactose has to enter into the bacteria with the help of enzyme permease. Are you getting that? Go with the thing what I'm speaking. Don't go with the marked here. What I'm speaking the answer, go with that right now, right? So what should be the answer here? With the help of enzyme permease. Are you getting that? So that is the lactopron question. Where permease is permitting the lactose to enter into the bacteria if we are giving the bacteria the lactose. Now coming to the question number 102. Lecithin, a small molecular weight that is an organic compound found in living tissue, is an example of. This is the table given in the biomolecules chapter. There you will find, and even in the formula of the structure, you will find all these structures of the phospholipids. The lecithin given over there, it is a type of the phospholipid. So you have to go with the third option with this question, right? Now coming to the match, the list 1 with the list 2 we have to go. This question is from microbes in human welfare, right? Let's take on the list 1, then match with the list 2. Clostridium botulicum. This you can correlate by the butyric acid, right? Saccharomyces cerevisiae. This is a yeast which helps in the production of the ethanol. Trichoderma polysporum. This is helpful for the formation or secretion of cyclosporin. We can extract from there. Then Streptococcus species. This is for streptokinase enzyme, right? What we are getting? 3142. 3142 means fourth option you have to go with this, right? So this 3 question in the starting is from botany. Now 104 question is based on zoology. Uma ma'am will take care of this.
Okay, next question is equation of Weller's Pearl logistic growth dn by dt is equal to Rn k minus n by k. In this formula, R is the intrinsic rate of natural increase, N is the number, that is size of the population, and K is carrying capacity. Carrying capacity is maximum number of organisms that can be supported by habitat in the given ecosystem. Okay, so correct answer for this question is carrying capacity.
Okay, coming to the next question on the botany, 105. Here they are asking hint 2, means based on restriction enzyme they are asking. Always cut the DNA molecules at a particular point called recognition sequence, and it consists of, means how many base pair that palindromic sequence of hint 2 is made up of. It is made up of 6 base pair, GTC, GAC. That will be the sequence of that, and this is for the 6 base pair. Mostly the recognition sites or the sequences having usually 6 base pair. Hint 2 is one of that which is having 6 base sequence here, right?
Also use different color because most of them are black. Choose.
Okay, now coming to the next question. What is that? 106 here. Formation of interfascicular cambium from fully developed parenchyma cell is an example of. when you will see this point, no, when you are having this interfascicular cambium, can I say this is the secondary meristem here right now? And they are saying fully developed. It is developed from where? Fully developed parenchyma cell, means it is formed from primary permanent tissue. So what type of process it will be? It will be dedifferentiation process, right? Dedifferentiation, right? So what option we need to go with this? We have to go with the fourth option, right? So 106 S3 code, you have to go with the fourth option, right? Now coming to the next part, inhibition of the succinic dehydrogenase enzyme by malonate is a classical example of, see, this is the name of the enzyme they have given over there, succinic dehydrogenase, and malonate they have given as an inhibitor, right? So it is competing with succinate, which is a substrate over there and usually bind with the succinic dehydrogenase to form its product. But right now, if this inhibitor is going to bind instead of the substrate, then they are going to inhibit the action of succinate dehydrogenase. This is a sort of competitive inhibition. So 107 question, you can go with the fourth option.
Next question, list of endangered species was released by. Actually, this is published in Red Data book, and Red Data book is published by IUCN. IUCN, International Union for Conservation of Nature and Natural Resources. So IUCN publishes a data list in which the list of endangered species is given, right? So IUCN is the correct answer for that question.
Okay, then after this, question number 109, this is based on cell cycle cell division. Spindle fibers attached to kinetochore of chromosomes during this direct line and even during the process, they are going to bind with the kinetochore in which phase? Metaphase, right? Spindle formation started in the prophase, but it started attaching to the kinetochore in the metaphase. So 109, third option is correct over there. Now coming to question number 110. How many molecules of ATP and NADPH are required for every molecule of carbon dioxide fixed in Kelvin cycle? Means you have to take for one cycle only. That means you have to go with the how many ATP molecules? 3 molecules of ATP. NADPH will be 2 over there. Now, when you check the option, you will find this thing in the first option, 3 molecules of ATP and 2 molecules of NADPH. So, 110, first option is correct here, right? Now, coming to the next question, 111. Let's check that. Match the list 1 with the list 2. So, here, when you will see here, 2 or more alternative forms of gene, that means we are talking about the alleles. You have to go with the 3 here, right? Then cross of F1 progeny with homozygous recessive parent, means we are discussing here by the test cross by which you can identify the genotype of dominant phenotype. Then cross of F1 progeny with any of the parents, that is a back cross, right? Test cross is even a back cross, but right now if you are saying homozygous recessive parent, then it is a test cross. Any of the parent, then it is a back cross. Number of chromosomes set in a plant or any organism, if you are talking about, that is its ploidy, so you can match it with 2, means 3, 4, 1, 2, means you have to go with which option right now? Fourth option, 3, 4, 1 and 2, right? Now next question, 112. Again based on list 1 and list 2, we have to match it. Like in case of Rhizopus, by which thing you have to match it? Its common name is a bread mold, you have to go with the 3 here. Astilago, the common name is a smut fungus. Then Puccinia is a rust fungus, you can match it with 4 right now. Agaricus is a mushroom, common name will be mushroom. So 3, 2, 4, 1. 3, 2, 4, 1. 3, 2, 4, 1 that you will find in option number 2 here. 3, 2, 4, 1, right? So question number 112, answer will be second option. Now coming to the next, given below are 2 statements. Let's check on statement 1. Chromosome become gradually visible under light microscope during lactoprotein stage. Very right, direct statement from your NCERT as well. Then the beginning of diplotin stage is recognized by this solution of synaptoneval complex. Even that is right, because after that dikinesis will happen where terminalization happen, but right now In diplotin, the beginning is happen by the recognition of dissolution of synaptonymal complex. Means first and second both are correct. So you have to go here with the second option, right? So 113, second option is correct. Now let's check on the next. 114. Here they are asking this diagram based question. Here they have given you the seed view of the maize and asking you identify the part of the seed from the given figure which is destined to form the root. This is a key point here. When the seed germinates, right? So you know that root is germinating from the radical part. So you have to go with the C here. As per your diagram, even in the labeling of +1, +2 book, you will find the C is the radical part, whereas the D part is a coleorhiza, right? So here you have to go with the C, means fourth option you have to go with that. C is there, right? Then after this, the question number 115. This is based on your genetics part. They are asking you here, giving you the example of seed color. Black is going to be the seed color with the dominance homozygous and heterozygous. And we have to cross it. Obviously, the recessive is BB they have given, which is a white seed here, right? Just they are assuming that thing and we have to go with the question. In order to find out the genotype of black seed, with which of the following genotype will you cross it? Because can you see here, the genotype now can be this or can be this. How we can check whether it is a homozygous or heterozygous dominant? By any of the, you can say this of the thing, you have to cross it, back cross, or you can say you have to test cross with the homozygous recessive parent. Which is the homozygous recessive parent? Like BB you have to take. This only they have given the, in the introduction, the type of the genotype and the phenotype what we are taking. So based on that just introduction given, to check its genotype of dominant phenotype, you have to back cross, or you can say test cross very correctly with the homozygous recessive parent BB, right? So which option you have to opt it? That homozygous recessive, right? BB will be your answer here. Now coming to the next question number 116. Here they are asking bulliform cells are responsible for which thing? You know that bulliform cells are present on the upper epidermis of the monocot leaves, right? If the water is plenty, then it will become turgid, it will open the leaf. When the water is less, the cell becomes flaccid and it will curl that leaf, right? So means they are working in the monocot leaves. Right now, if you see the option, like second option is most appropriate to pick it and correct, obviously not most appropriate, it is correct, rest all are 100% wrong. Like first they have given providing large space for storage of sugars, it is an empty cells, how can sugars be there? Then protecting the plant from the salt stress, even not as a thing, this is the helping in overcome the stress of the water. Increased photosynthesis monocot, not for the job of this. So you have to go with the second option, inward curling of leaf in the monocot's leaf when the water stress is there, right? Now coming to the question number 117. In this question, you will find some statements have been given. Let's sort it out, because the demand of question, they are asking which one of the following can be explained basis on the Mendel's law of dominance. And I'm directly reading the demand of the question. What is that? Choose the correct answer from the options given below. Now we are clear with the thing, what we have to go with the statements. Let's check it. Out of one pair of factor, one is dominant and the other is recessive. This is going to be the right, because Mendel law of dominance they are talking about. Then after that, alleles do not show any expression. They will show the expression. If it is a homozygous dominant, then it will show that. And both the characters appear as such in F2. So this is wrong based on the statements given here. Then factors occur in pairs, normal diploid plants. That's right. They are in paired. If I am taking capital T as one of the allele, it should have the small t as well, right? Okay, just a moment. Okay, so there is some instruction for the pencil size. I will use that more in that case. Okay, let's check the next one. So here choose the correct answer they are saying. Let's check for the C once again. Factors occur in pairs in normal diploid plants. Obviously, diploid plants will be having a pair of alleles, right? Either it is a homozygous, heterozygous, so it will be having pair. This is right. The discrete unit controlling a particular character is called factor. This is even right. The expression of only one of the parental characters, right? Parental character 1 will be shown by monohybrid cross. Even this is right. So you have to go with A, C, D, E. A, C, D, E are correct. So third option you have to go with. 117, third option, right? Now stick on 118th. The cofactor of enzyme carboxypeptidases. Carboxypeptidase, they need the cofactor which is metal ion form. Binding with that, with the coordinate bonding over there. And what is that thing? That will be the zinc. Right? Direct line from your NCERT, example even from your NCERT they have taken for the cofactor, right? Fine. Okay, then after this, transcription unit in DNA is defined primarily by the 3 regions in DNA, and these are with respect to upstream and downstream. Downstream end? What is the sequence of that? This is from transcription unit. So we have to consider for transcription unit right now. There you will find there will be the promoter will be started on the upstream, and downstream it will be having for the terminator, like this one, right? I hope so the pencil size is clear to everyone. Fine. Okay, so here is a promoter here, and here will be the terminator, and this is the structural gene, right? So this is upstream and this one is a downstream, right? So you have to go from, as a demand of question, that is from upstream towards the downstream respectively you have to go. Then promoter, structural gene and the terminator, means first option is correct over there, right? Now next question is from zoology.
Tropical regions show greatest level of species richness because. First statement, tropical latitudes have remained relatively undisturbed for millions of years. Hence, more time was available for species diversification. This is the correct reason for high diversity in tropics. And the second one, tropical environments are more seasonal. This is wrong. They are less seasonal. Seasonal changes are less prominent in tropical region. And more solar energy is available in tropics. Because tropics are close to the equator, amount of sunlight available or solar energy available is more. So definitely that is a correct one. Constant environments promote niche specialization. So definitely this niche specialization is promoted by constant environments. So D is also correct. And tropical environments are constant and predictable. Yes, that is also true. That means A, C, D, E. Correct answer, the second option. Only B is wrong. It should be less seasonal, not more seasonal. Right? Okay then.
Okay, now coming to the question number 121. Here what they have given. That given below 2 statements. Statement 1 is what? Parenchyma is living. That's right. No doubt in that. And collenchyma is dead tissue. This is wrong part of this statement. Are you getting that? So can I say overall this statement is wrong here? Then statement 2, gymnosperm lack xylem vessels. This is right. But the presence of xylem vessel is a characteristic of angiosperm. This is right. So what can I conclude from the full discussion here? Can I say the first statement is wrong due to this part, collenchyma is living tissue, right? And statement 2 is correct. So if I am saying based on the false and the true, then statement 1 is false and statement 2 is true. So we have to go with the first option, right? Now coming to the next question, 122. That is the zoology question.
The type of conservation in which threatened species are taken out from their natural habitat and placed in special setting where they can be protected and given special care is called. Actually, that is ex situ conservation. Correct answer for that is ex situ, because the species are taken out of the natural habitat and placed in protected environment. So it is ex situ, but that option is not there. In situ is given. Anyway, in situ and ex situ are two methodologies used in biodiversity conservation. So you can go along with third option, biodiversity conservation.
Okay, now coming to the next question number 123. This is based on photosynthesis. Which of the following are required for the dark reaction of the photosynthesis, right? So light and chlorophyll are for the light reaction, not for the dark reaction. Hint for us in the question is a dark reaction, right? Light and photosynthesis, that is not required for the dark reaction. We have to go with the carbon dioxide, ATP, NADPH. ATP, NADPH are the product of light reaction, even that is required. So we need C, D, E. Means which option is correct based on that? Fourth option is correct based on this, right? Now coming to the next question, 124. This is based on biotechnology. Here they are asking statement 1 and statement 2. Regarding that, we have to opt the option here. Statement 1, they have given Bt toxin are insect group specific and coded by a gene like Cry1AC. Cry1AC, you know very well, this is one of the gene which is secreted, or these are having that gene in the, or where these genes are, these are present in bacteria and secreting for a toxic chemical which is killing a specific insects, right? So this is correct statement. Then next statement 2, Bt toxin exists as inactive protoxin in Bacillus thuringiensis. This is right. However, after ingestion by the insect, the inactivated protoxin get converted into active. Up to here is right. Formed due to acidic pH. This is wrong part, right? This hidden word is wrong here. That is the alkaline pH here. Are you getting that? In the insect gut. So can I say here statement 1 is right and statement 2 is wrong, or based on that true and false? Here I can go with statement 1 is going to be true and statement 2 is false. Fourth option we have to go, right?
Next question. These are regarded as major causes of biodiversity loss. And you know about that evil quartet, the 4 major causes for biodiversity loss. Among those, first one, overexploitation, it is there. Co-extinction, one of the reasons. Mutation is not related to that. And habitat loss and fragmentation. This is one of the major cause for biodiversity loss. And migration is not a specific reason. So if you go along with this, correct answer is A, B, D only. So this is the correct answer for that.
Fine. Now coming to the next question, question number 126. Here this is diagram based question. In the given figure, which component has thin outer wall and highly thick inner wall? Can I say this is based on this guard cells which are guarding the stoma over here? So it is having very thick towards the stoma side and thin towards outer side. See the labeling, what they have given to that C here. So what option goes with that? Second option is correct, right? Then next question number 127. They are asking here the capacity to generate a whole plant from any cell of the plant is called. That process or that capacity, that you call it as totipotency. The process they are not asking process, that capacity to generate the whole plant from any cell of the plant, that you call it as totipotency. Somatic hybridization is simply the fusion of protoplast, that is not for this concept. Then micropropagation is the process to multiply that in bigger number, where totipotency will help, this capacity will help to do so. Differentiation is not here. In the tissue culture, we will work on de-differentiation process. So ultimately your answer will be second option. Now coming to the next question, 128, based on the genetics again, pink flowered snapdragon plant was crossed with the red flower snapdragon plant. If it is pink color, can I say for the pink color, you have to take the heterozygous one, being it is showing incomplete dominance, and we have to cross it with the red plant, a snapdragon plant, what type of phenotype is or are expected in the progeny? Can I say it will form two types of gamete, capital R and small r? This will form only one type of gamete. Type, I am saying type of gamete will be one. If this is fusing with this, can I say we will get red? If this with this, then can I say we will get pink? Now get back to the options. Here, what they are asking, the pink flowered snapdragon plant was crossed with the red flowered snapdragon plant. What type of phenotype is on our expected in the progeny? First option, red, pink as well as white flower. Can you see there is any white flower? Not at all. So we cannot go with that. Only red flowered plant, even that is wrong. Third, red flower as well as pink flower, that is right. Only pink flowered plant, again this is wrong, right? Now coming to the fields, ultimately 128, third option is correct. Okay, now coming to the next question, match the column once again, list 1 with the list 2. Here, nucleolus, can I say this is a site for active ribosomal RNA synthesis? You can match it with 3 now. Centriole, can I say it is organized like the cartwheel here? Second, you have to match it with. Leucoplast, for storing nutrients. Can I say you have to go with the 4 now. Golgi apparatus, site of formation of glycolipids, right? You can match it with 1. Can I say 3, 2, 4, 1 now? 3, 2, 4, 1. So means our answer here is going to be 3, 2, 4, 1 means second option is right for question number 129. Now coming to the next question, this is again diagram based on the morphology part. Identify the type of flowers based on the position of calyx, corolla and androecium with respect to the ovary from the given figure A and B. When you will see this diagram in your NCERT, the central two diagrams, right, that is indicating that part, means we have to go with perigynous, first one, and even second one you have to go with the perigynous condition over there. Means you have to select for the first option here with the perigynous, and perigynous, let me do check that even as per your NCERT book. So fine, so you have to go with the perigynous and perigynous. So first option you can go with that, because here you will find the diagram both they have given for perigynous. Before that they have given for hypogynous and epigynous like that. So A and B both are indicated for perigynous, right? Now coming to the next part, identify the set of the correct statement. This is based on your chapter sexual reproduction in flowering plants, right? So here the first statement they are saying the flower of Valsneria are colorful and produce nectar. Why it have to do so? Nothing like that, because they have to pollinate with the help of water. They need not to be attractive and need not to form the these nectar over there. So this is wrong statement. The flowers of water lily are not pollinated by water, because it is pollinated by insect or by the wind. Then most of the water pollinated species, the pollen grains are protected from wetting. This is right. Then pollen grains of some hydrophytes are long ribbon like. That's right, because they are in the water. For that thing it should be narrow and will be ribbon like, make them too persistive there. In some hydrophytes the pollen grains are carried passively inside the water. This is even right. So which statement are correct? B, C, D, E. If you go with this thing, right, B, C, D, E, right, you will find here the option number 4 will be correct, right? So A, C, D, fourth option is correct for this, right? Okay. Now question number, next you have to go with next question number 132. Auxin is used by gardeners to prepare weed-free lawns, but no damage is caused to the grass as auxin. What is the case here? Can help in a cell division in grasses to produce growth. Nothing related to that. Then promotes apical dominance, no doubt. It is a job. It is not a job of auxin. It is promoting, no doubt. It is promoting the apical dominance, but right now it is not related to the weed-free lawns, right? So promotes the abscission of mature leaves only, even not correct. Does not affect mature monocotyledonous plant. This is right. right? It is going to eradicate dichotomous wheat, but it will not affect the main crop, or you can say your lawns having grasses means monocots. So can I say it does not affect the mature monocotyledonous plants? So this is the correct thing. You have to go with the question demand, right? So 132, fourth option. Now coming to the next question, number 133. What is the fate of piece of DNA carrying only gene of interest which is transferred into an alien organism? We kept it in an alien organism, the gene of interest. The piece of DNA would be able to multiply itself independently. It cannot happen. It cannot multiply independently over there until and unless it is not incorporated with the host DNA, right? It may get integrated into the genome of the recipient. That is right. They may independently, they get integrated into genome of the recipient. That's fine. If it is going to integrate it into the genome, only then it can multiply by using the various thing ORI as well of that. It may multiply and be inherited along with the host DNA. That's why we can get the multiple copies of that. Then the alien piece of DNA is not an integral part of the chromosome. It's somewhere it is right as well as a wrong type of statement. But if you go with the options, they have given majorly the combination of two only, right? So I'm keeping it wrong. The alien piece of DNA is not an integral part of the chromosome. Somewhere it is right if I'm taking, because you have kept it from outside, right? So on that cases we can take it as a wrong. Even if we go with the combination with BC, you don't have any third combination. Why I'm taking wrong? Because alien piece of DNA is not a part of the main recipient chromosome over there, right? Then it shows ability to replicate here. So here we have to go with, they cannot independently show the ability of the replication because they are not having ORI at that moment. So they are asking for correct answer with the options which are correct for that. B and C are correct, means you have to go with the fourth option. The next question based on biological classification on the kingdom fungi. Which one of the following is not a criterion for classification of this organism, fungi? Fruiting body, yes, this is a criteria. Morphology of mycelium, yes. Mode of nutrition will not, because all are heterotrophic, right? This is a type of kingdom which is special based on such type of thing. Now monotony is there in their mode of nourishment. They are heterotrophic, mainly the saprophytic. Then is what mode of spore formation? Yes, even that is the criteria. So what is odd one out? Third option. So you have to go with that, right? Then which of the following is an example of actinomorphic flower? When you see the option, first given Sesbania. Sesbania is from which family? This is from the Fabaceae family. It is having zygomorphic type of you can say the symmetry, so you cannot go with that. Datura, yes, you can go because this is from Solanaceae family, showing the actinomorphic type of the symmetry. Acacia is again Fabaceae family. This is a zygomorphic. Again, you will not take it. Fourth one, to check it, just a moment. I have to go with that now. Find just. So here, let's go with the options here. Again, I am repeating. Sesbania, we are not taking. Zygomorphic. Datura, actinomorphic. Yes, we can go with that. Acacia, again Fabaceae family, zygomorphic. We will not go with that. Piscium, again from the you can say Fabaceae family, again zygomorphic. You should not take. So what is an answer for question number 135? That will be the second option, right? So focus on this question right now. This is a little bit shuffle, right? 135 question. Clear? Okay, now coming to the next question, 136. Again, match the column. We are entering into the section B now. I hope so you have selected very nicely and feasibly what type of question you know, and based on that, you opt and answer that. Now, question number 136, match the list 1 with the list 2. Rows, by which thing I can match here? Rows I can match with the perigynous flower. Just now we have taken its diagram. Then P is what? With the marginal placentation. Fourth, we can go with that. Cotton, with the aestivation we can match here with the twisted aestivation. Mango with the type of fruit, that is a drupe, right? So 2, 4, 1, 3. 2, 4, 1, 3 means second option you have to go with this, right? So question number 136, second option, right? Then coming to question number 137, diagram based question again from sexual reproduction in flowering plant. Identify the correct description about the given figure. So you remember this is for the corn, they have shown the male female on the same inflorescence. So based on that, this is a feature of wind pollinated inflorescence where the male are even projecting out. So if you go with the option, the most you can say, I must say the correct option will be the second, wind pollinated plant inflorescence showing flower with well exposed stamens. Upper side usually the male part are there, lower side female is there, so it is even exposed. So this is a feature of wind pollinated, not of water pollinated, right? So question number 137, second option is correct here. Now coming to next question number 138, again match the column. This is based on your chapter biomolecules. So here GLUT4, what it helps in, it enables the glucose to transport into the cell. This is a type of protein, this is its function. So A you can match with the 4. Insulin, this is working as an hormone, again is a protein, so here first you can match it with that. Trypsin is an enzyme, you can match with the 2. Collagen is working as intracellular ground substance, you can match with 3. So 4, 1, 2, 3. 4, 1, 2, 3 means second option you have to go with. Fine. So question number 138, second option. Now question number 139, plant growth and development from their hormones they are taking. Spraying sugarcane crop with which of the following plant growth regulator increase the length of stem, thus increasing its yield? That will be gibberellin. Help to increase the internodal length and sugar production will be there. This is the role of the gibberellins here. So 139, third option. Then question number 140, which of the following are fused in somatic hybridization involving 2 varieties of plant? They are asking somatic hybridization, right? So means we are taking 2 plant cells and we are removing their cell wall and their protoplast we make them to fuse, right? So here what thing will fuse over there? Protoplast. I again repeat, where we are making tick, focus on that answer right now, right? So question number 140, fourth option is correct, right? Then question number 141, they have given 2 statements, statement 1 and 2, and based on the option we have to mention false or true. Now first statement, in C3 plant some oxygen binds to ribosco, hence carbon dioxide fixation decrease. That's right, because ribosco now working as oxygenase, then obviously the photosynthetic rate will come down. Next statement 2, in C4 plants mesophyll cells show very little photorespiration. There is no ribosco in the mesophyll cell in the case of C4 plants. While bundle sheet cell do not show photorespiration, they little bit can show but maybe negligible, but they cannot show in the mesophyll cell being ribosco absent. So we can go with the wrong statement. Can I say first is true and second is false, means you have to go with the fourth option, right? Now coming to the next question, 142. Read the following statement and choose the set of the correct statements, right? We have to go with the correct now, but hint is what? In the membrane, in the members of the Phaeophyce, brown alga, right? So they are saying asexual reproduction occurs usually by biflagellate zoospore. This is fine. But sexual reproduction, now B, they are saying sexual reproduction is by oogamous matter, right? But only this is not correct. That's why this statement will be wrong. Then stored food in the form of carbohydrate, which either be mannitol and laminarin, right only. The major pigment found in chlorophyll, A, C, carotenoid and xanthophyll, right only. C is there in the brown alga, carotenoid, xanthophyll, that xanthophyll specially for giving brown color, that's right. Vegetative cells have cellulosic wall, that's right, usually covered only on the outside by gelatinous coating algin, even this is right. So which statements are right? A, C, D, E, means you have to go with the fourth option for question number 142. Now coming to the next question, match the column, that is from zoology.
Robert May, he estimated the global species diversity at about 7 million. So it should be third. Alexander von Humboldt, he explained the species area relationship. So it must be 1. Paul Ehrlich, he explained or proposed this rivet popper hypothesis. 4. David Tillman, long-term ecosystem experiments using outdoor plots. These experiments were conducted by David Tillman too. So, correct answer is 3142. Third option. Okay, the next one.
Okay, now next question is again match the column, list 1 and list 2. Frederick Griffith, we can match it by the transformation process, that is the third one. Then that is Francois Jacob and Jack Monod. This is based on Lacapron. You can match it by 4. Then is the Hargobind Khurana. We can match it by genetic code, the work given by him there. Mazelcell and Stahl for the semi-conservative mode of DNA replication. You can go with the 2 here. 3, 4, 1, 2. 3, 4, 1, 2 means third option you have to go with that, right? Then question number 145. Again match the column from the morphology chapter. Here Monoadelphus, we can match it by China rose, fourth option. Then Diadelphus, you can match it by the P for the Fabaceae family. Polyadelphus, you can match with the citrus first. Then Apiphyllus, we can match with the Liliaceae family member, lily here. So 4, 2, 1, 4, 4, 2, 1, 4. 1, 3, sorry, 4, 2, 1, 3. That means the second option you have to go, right? 145, we have to go with the second option. Now coming to the 146, the DNA present in chloroplast. Chloroplast is having DNA, that's why you call it as semi-autonomous cell organelle. So in that, the DNA will be circular and double-stranded. So third option you have to go with, right?
147. In an ecosystem, if the net primary productivity of first trophic level is 100x per square meter kilocalories per year, what would be the GPP of the third trophic level same ecosystem? Actually, as per 10% rule, from first trophic level to second trophic level or from second to third, any trophic level, only 10% is transferred. It's called 10% law, right? And as per that, you can go for the third option, but I find a mistake in the question itself. Actually, this gross primary productivity is observed only in the first trophic level. First trophic level is occupied by producers, and the productivity in producers or first level is called as primary productivity, which is again of two subtypes like gross and net primary productivity. Second trophic level onwards, the members are consumers. So, the productivity in consumers is called secondary productivity, not primary productivity. So, there is a mistake in the question itself. They should have asked secondary productivity in the third trophic level, not gross primary productivity. So, let us see what they will do with this particular question. Okay, because there is a mistake in the question itself. It is not gross primary productivity at the third trophic level. Okay, then we proceed with the next question.
Okay, so next question 148. This is based on your chapter, that is respiration. Identify the step in a tricarboxylic acid cycle which does not involve oxidation of the substrate, right? They have given for isocitrate to alpha ketoglutarate glutaric acid. Here there is a formation of NADH, so means oxidation happen. Malic acid to the oxaloacetic acid, even here the NADH formation is there. Succinic acid to the malic acid, again oxidation happens here. So even here we cannot take it. Succinyl coenzyme to succinic acid, there is a formation of ATP, not NADH or FADH here. So here answer will be the fourth option, succinyl coenzyme to succinic acid, ATP formation is there. Rest of the cases you will find H plus will be picked up by the coenzyme like NAD plus or FAD plus. So we should not go with 1, 2 and 3. You have to go with the fourth option. Now coming to the next 149. Which of the following statement is correct regarding the process of replication in E. coli? Here they are asking for the process of replication in the bacteria E. coli, right? So here they are asking for the replication. You know that when the replica is going to be formed on the template, the new replica, the formation is from 5 prime to 3 prime, and this is done by DNA polymerase, not by RNA polymerase, because we are discussing about replication process here, right? So here first statement they are saying the DNA dependent DNA polymerase catalyzes the polymerization in 5 prime to 3 prime direction. This is a right, but let's wait for the other things as well. The DNA dependent DNA polymerases catalyzes polymerization in one direction, that is 3 prime to 5 prime. This is wrong. Then the DNA dependent RNA polymerase, we should not talk about RNA polymerase. That's why this statement will be wrong. The DNA dependent DNA polymerase catalyzes polymerization in 5 prime as well as 3 prime to 5 prime. This is wrong. So what is the final answer correct for that? First option is correct, right? Okay, now coming to the next question, number 150. What is that given? Again, the match the list 1 with the list 2. The list 1, they have given citric acid cycle. Where it happens? It is happening in the case of mitochondrial matrix. You have to match it by the 2. Then the glycolysis, where it happens? In the cytoplasm. 1 you have to take. Electron transport system. It is happening in the inner mitochondrial membrane. You have to go with the 4. Then proton gradient in the intermembrane space of the mitochondria, it will develop. So you have to go with 2143. 2143 means third option. Just a moment, before getting into the next zoology part, I want to go on the question number 131 again. 131, let's check, right? Here 131, this question. Right, again we are doing on this question. Have a look here. I again repeat, we are back on the question 131 before entering into the zoology part, right? Okay, let's see this question once again. Identify the set of correct statement. First they have given the flower of Welsonaria are colorful and produce nectar. This is not correct, because being it is going to pollinate by the water, not by any biotic agent. So that's why this is wrong statement. The flower of water lily is not pollinated by water. This is pollinated by the wind or the insect. That's why this is a correct statement. The most of the water pollinated species, the pollen grain are protected from wetting. That's right, because it is having mucilaginous sheet over there. Then pollen grain of some hydrophytes are long and ribbon-like. Even that is a correct. In some hydrophytes, the pollen grain are carried passively inside the water. This is even right. You have to go with the correct statement, means you have to go with the B, C, D, E. I repeat here, first option is correct here, right? Please do check question number 131. We have to go with the first option. Clear? Now we are proceeding for the zoology section here.
Next question 151. Given below are two statements. First one, in a nephron, the descending limb of loop of Henle is impermeable to water and permeable to electrolytes. The statement is wrong because descending limb of loop of Henle is permeable to water. Ascending limb is impermeable to water, but descending limb is permeable to water. So this is wrong statement. And second statement, proximal convoluted tubule is lined by simple columnar brush border epithelium and increases the surface area for reabsorption. No doubt, the epithelium with brush border increases the surface area, but it is not columnar epithelium. It should be cuboidal, not columnar. So, both the statements are wrong. So, correct option would be third one. Both statement 1 and statement 2 are false, right? Okay, then the next question 152. Again, assertion reason question. Assertion, FSH acts upon ovarian follicles in female and Leydig cells in male. There is a mistake here. FSH acts on Sertoli cells, LH acts on Leydig cells. So, in the case of male, FSH acts on Sertoli, not Leydig. That's why assertion is wrong. And coming to reason, growing ovarian follicles secrete estrogen in female, right? And while interstitial cells secrete androgen in male human being, interstitial cells are nothing but Leydig cells. That means the second statement or reason point is correct. So, correct option would be first one. A is false and R is true. Okay, then the next question, 153. Which one of the following factors will not affect Hardy-Weinberg equilibrium? Hardy-Weinberg equilibrium is affected by mainly 5 factors like genetic recombination, genetic drift, gene migration, natural selection, mutation, all these affect the Hardy-Weinberg. So, factor which does not affect the equilibrium is constant gene pool. And you know very well that Hardy-Weinberg equilibrium actually discusses about constant gene pool and the frequencies remaining constant generation after generation. Okay. So, that is about 153. First one is the correct option. Next, 154. Which of the following are autoimmune disorders? Myasthenia gravis is autoimmune disorder. Rheumatoid arthritis, textual example is clearly given NCERT. Gout is due to accumulation of uric acid crystals in the joints, not autoimmune disorder. So, this is wrong. Muscular dystrophy is genetic disorder, not autoimmune. So, that is also wrong. Systematic lupus erythematosus, this is another autoimmune disorder. So, correct option would be A, B, E. Right. Third option is the correct one. Okay. Then, next question. Given below are some stages of human evolution. Arrange them in correct sequence, past to the recent. So, chronological order is given. The first form is Homo habilis. After Homo habilis, Homo erectus appeared, followed by Homo neanderthalensis. C, and the last one, Homo sapiens, that's the modern man. So, A, D, C, B, first option is the correct one. Okay. And the next question, again statement. Presence or absence of hymen is not a reliable indicator of virginity. Yes. Statement is true. And the second one, hymen is torn during first coitus only. This word only makes it wrong because hymen can be torn due to various other reasons also, due to physical activity or usage of tampons and all this. So many reasons are there. So as they specified this word only, it becomes wrong. So statement 1 is true and statement 2 is false. That means you have to go along with the fourth option, right? Then next question, matching. This is about contraceptives. Non-medicated IUD, it is Lippis loop, so 3. And then copper releasing IUD is multi-load 375, so 1. Then hormone releasing IUD LNG 20, so fourth one. And implants contain progestogens on progestogen estrogen combination, so second option. That means 3, 1, 4, 2. First option is the correct combination, right? Next question, 158. Consider the following statements. Annelids are true coelomates, right? They have schizocoelom and the true coelom. Poriferans are pseudocoelomates. Pseudocoelomate phylum is Ascalmenthes. Porifera have no coelom, so this is wrong. Ascalmenthes are acoelomates. Ascalmenthes are pseudocoelomates. That means this is also wrong. Then D, Platyhelminthes are pseudocoelomates. Platyhelminthes are acoelomates. So all these remaining options are statements are wrong. So the only correct one among the four, first one, so A only, third option is the correct one. Okay. Then the next question, 159. Which of the following factors are favorable for formation of oxyhemoglobin in alveoli? Actually, oxyhemoglobin in alveoli is favored by high PO2, low PCO2, low H plus and low temperature condition. So, in the first option, low PCO2 may be considered, but high temperature makes it wrong because temperature must be low to facilitate oxyhemoglobin formation. High PO2 is correct, but high PCO2 is wrong. Okay, and high PO2 true, less H plus concentration. So third option has both factors appropriate. Fourth option, low PCO2 is correct, but high H plus is wrong. So third is the correct set of factors which promote oxyhemoglobin formation. Then next one, 160 question, matching. Expiratory capacity. Expiratory capacity is TV plus ERV, tidal volume plus expiratory reserve volume. That means second one. Functional residual capacity, FRC, is equal to ERV plus RV, expiratory reserve volume plus residual volume. That means fourth one. Then vital capacity is TV plus IRV plus ERV. That means expiratory reserve, tidal volume, first option. Then inspiratory capacity is nothing but TV plus IRV. So TV plus IRV, third one. So correct combination 2, 4, 1, 3. Second option gives the correct combination. Okay. Next question, 161. Yeah, you are given a figure here. All three types of muscles are represented, and here this is a skeletal muscle, these are smooth muscles, these are cardiac muscles. Okay. So let us check the options given. Involuntary, nose tip. Nose tip actually has hyaline cartilage, and that too, it's not a correct muscle. Skeletal muscle attached to bone, cardiac present in the heart, but these are not correct. Smooth toes, skeletal legs, cardiac heart is given. Skeletal muscles are seen in triceps. Triceps and biceps are the muscles present in the hand. So that is a skeletal muscle. And smooth muscles are involuntary muscles present in the alimentary canal, stomach, intestine, etc. So second B is correct. Cardiac is present in the heart. So third option gives the correct combination. Okay. Fourth option, smooth muscle is given in heart. That is wrong. So third one gives the correct combination. Okay. Then the next one, 162. Flippers of penguins and dolphins are example of. This is a textual example. It's clearly mentioned in NCERT. Penguins are the birds, dolphins are the mammals. So they belong to two different classes, and both of them have developed flippers which help them to swim in the same habitat. So different organisms developing similar characters to live in the same habitat. It is nothing but convergent evolution. So fourth one is the correct option. Okay. Then the next one, matching question. Typhoid. Typhoid is caused by Salmonella typhi, which is a bacterium. So that is the fourth option. Then leishmaniasis. Leishmania is a protozoan. Leishmaniasis is caused by leishmania, which is a protozoan. Ringworm is fungus. Ringworm is a disease caused by fungi. And filariasis is caused by Wuchereria, which is a nematode parasite. So correct combination. Even though you know the first one, typhoid is caused by bacterium, you can mark the correct option. 4312. Okay. The next one, 164. Following are the stages for pathway, conduction of action potentials through the heart. Actually, the potentials are generated in SA node first. From the SA node, they are received by AV node. From the AV node, AV bundle. From the AV bundle, right and left bundle branches, and finally through the Purkinje fibers spread in the walls of the ventricles. So correct option would be E, C, A, D, B. Second option. Okay. And then, yeah, next question will be discussed by Botany ma'am.
Okay, now match the list with list 2. This question, they are asking for the role of this enzyme. We have to match with the list 2. Here the lipase, they are going to break the ester bond here. So can I say here it can match with the 2? Just a moment. Lipase, we can match with the 2 right now with the ester here. Then B, nuclease, that is going to be the match with the 4, right? Phosphodiester. Protease, we can match with the peptide bond. and amylase we can match with the glycosidic bond here, right? So it is going to be 2413. 2413 means we can match it with the fourth option, right? We can match it with the fourth option, right? Somewhat clarity maybe is not there. I am writing here. We can match with the fourth option. Question number 165. 2413. You can match with the fourth option. Fine. Now coming to next question, 166 here. Here this is related to biotechnology again. In the biotechnology, they are asking the role of the bioreactor. What thing is incorrect regarding that, right? So here the bioreactor have an agitator system and oxygen delivery system and foam control system. That's right. Then after this, a bioreactor provides optimal growth condition for achieving the desired product. Even that is right. Then most commonly used bioreactor are stirring type. That's right. Then fourth is what? Bioreactor are used to produce a small scale bacterial culture. This is wrong. We are using the bioreactor for the large scale production. And in the question, the demand they are asking for incorrect. So as per, we can go with the fourth option as an answer here. 166, fourth option. Now coming to question number 167. Here, this is biotechnology question. They are asking the TI plasmid of Agrobacterium tumefaciens. TI stands for what thing? That is the tumor inducing plasmid. T stands for tumor, I stands for inducing plasmid, already given. So tumor inducing plasmid. Fourth option will be correct over there. 167, fourth option is correct. Now coming to the next question.
Next question is again matching question. Common cold is caused by rhinoviruses. So if you know that combination, then you can select the correct option. With the first one itself, we can choose the correct option. Common cold is caused by rhinoviruses. Hemozoin is a toxic substance produced by Plasmodium, which causes malaria. And viral test is important for, or it helps to diagnose the typhoid. Allergy is caused by dust mites. So common cold is rhinovirus, then hemozoin is Plasmodium, viral test typhoid, and allergy dust mites. That means 3, 1, 2, 4. Fourth option is the correct one. Then next one, stages of cell division.
Okay, so for this thing, following are the stages of cell division. Choose the correct sequence. Now we have to go with the correct sequence here. When you see, you know that gap 1 is happening first during the interphase. So you have to go with the G1. Then after that, the synthesis will happen. Means the replication is a major thing going to happen over there. Then can I say for the G1 they have given E. Then for synthesis they have given for the C. Because we have to go with these things given the letters over here. So I repeat here. GAP1 phase is happening first, so E you have to take. Then synthesis phase due to the DNA replication, C you have to take. Then G2 phase, A you have to take. Then karyokinesis happen, then is a cytokinesis happen. ECADB, DCADB means first option you have to go with this question, right?
Next one, which of the following is not a natural or traditional contraceptive method? Waltz, coitus interruptus, periodic abstinence, lactational amenorrhea. These three are the methods of natural contraception. So the only possible answer is Waltz. Okay. And the next one, matching. Pleurobrachia is example for Ctenophora. So second one. Then radula is the rasping structure found in mollusca. So 1. Stomachord is characteristic of hemichordata. Fourth option. Air bladder is found in Osteichthyes. 3. So 2, 1, 4, 3. Third option is the correct one. Okay. Next one.
Now question number 172, that is the molecular basis of inheritance. Which one is the correct product of DNA dependent RNA polymerase to the given template? Means we have to talk about the transcription here, and template given, obviously the polarity here 3' to 5'. If we want to take the RNA, you can say the sequence on that. can I say it will be complementary to that? And even polarity will be 3' opposite to 5', right? So I am writing for you here, that is 5' to 3', right? So in that case, T is given first, then I take it as a A. Then after that, U will be there, right? Then after that, G, right? Then is for U, then is for A, C, then C, then G, then after that U, then is for one more U here, then is A, U, then after that A, then again more are there actually, GGU and AA, GU 3'. When you will check the sequence, that will make it nice here, GU, right? If you will check this part, you will find all the things given here in which option? That is in a second option. So here the answer for this question will be second option, right?
Next, another matching question. Alpha 1 antitrypsin is used as treatment of emphysema. So it is a third one. And Cry1AB, this is corn borer, and Cry1C, cotton bollworm. Enzyme replacement therapy, this is done for ADA deficiency. So even though you know these two, first and the last one, antitrypsin for emphysema, enzyme replacement therapy for ADA deficiency, you can choose the correct option easily. Okay. And then the next one, examples, common names are given. Pterophyllum is commonly called as angelfish. So third one, Mixin is hagfish, Pristis is sawfish, and Exocetus is flying fish. So correct combination is 3, 1, 2, 4. The third option is the correct combination. Okay. And the next one, 175. Which of the following is not a component of fallopian tube? Fallopian tube starts with infundibulum, followed by ampulla, and then isthmus. So these three are the parts of fallopian tube. Uterine fundus is the part of uterus. It's not a part of fallopian tube. So answer is uterine fundus. Okay, next one.
Okay, so this question is based on biotechnology, diagram based. The following diagram showing restriction sites in E. coli cloning vector PBR322. Find the role of X and Y genes. When you will see the diagram, they have given this as an X and this as a Y. And X, you remember, if it is for, this is for ORI, Y is for ROP. And if you will see the option, the third option is the right answer, because they have written the gene X means ORI is responsible for controlling copy number of the linked DNA, and Y for the protein involved in the replication of plasmid, which is for ROP. So question number 176, you have to go with the third option.
177, again a matching question. Fibrous joints, they are found in the skull and do not allow any movement. So third one, cartilaginous joints, they are present with adjacent vertebra, intervertebral disc, and they allow the limited movement. So first one, hinge joints, knee and elbow joints are hinge joints, they help in locomotion, they cause movement. So fourth one, and ball and socket joint between humerus and pectoral girdle, second one. So 3, 1, 4, 2 is the correct combination, that means first option. And the next one, which of the following is not a steroid hormone? Glucagon, it is given as the peptide hormone in NCERT, and cortisol. Cortisol is glucocorticoid, and all corticoids are steroid hormones. Testosterone, progesterone, these are sex hormones, and all these are again steroids. So cortisol, testosterone, progesterone are steroids. Glucagon is not a steroid hormone. And then the next question again matching. Axoneme is found in cilia and flagella. So second one. Cartwheel pattern, centriole, and then crista. Crista, everyone knows, I think, mitochondria, and then satellite related to chromosome. So 2, 1, 4, 3. First option is the correct combination. And then again another matching question. Cocaine extracted from Erythroxylum coca. So third one. Heroin. Heroin is formed by acetylation from morphine and it is extracted from papaver. Fourth one. Morphine is used as effective sedative after surgeries. So first one. And marijuana is obtained from cannabis sativa. So 3, 4, 1, 2. First option is the correct combination. In both sexes of cockroach, a pair of jointed filamentous structures called anal cerci are present on 10th chromosome. Actually, abdomen of cockroach has only 10 segments and the last segment, the 10th one, bears those filamentous structures, right? Next question.
So this question number, again based on the match, the list 1 with the list 2. They have given the substages of prophase 1. We have to match it by the specific characters. Like here, we have to go with the first of all diakinesis. Diakinesis is for completion of terminalization of chiasmata. Means we have to match A with 2. Then packitine. Packitine is for appearance of recombination nodules. Right? So we can match packitine with the 4 here, where recombination nodules help in the crossing over. Then zygotine is matched with the synaptic complex formation, due to which the homologous chromosome will remain close to each other. Then lactotine, here the chromosome look like threads only, right? So here you can match with the 3 because it is the first stage of the prophase 1. So here you can go with the 2, 4, 1, 3. 2, 4, 1, 3 means fourth option you have to go with.
Next question is assertion reason. Assertion. Breastfeeding during initial period of infant growth is recommended by doctors for bringing a healthy baby. Yes, it's true. And colostrum contains several antibodies absolutely essential to develop resistance for the newborn baby. Colostrum is a milk secreted in the initial days and it is rich in antibodies. These antibodies reach the baby and provide passive immunity, help the baby to resist various infections. So definitely this is the correct reason for this assertion. That's the reason why doctors recommend breastfeeding. Breastfeeding is said to be the best way of feeding a newborn baby. So this is the correct explanation. That means second option. And then another matching question. Down syndrome. Down syndrome is the trisomy of 21st chromosome. So third one. It matches the third one. Alpha thalassemia, gene causing alpha thalassemia is located on 16th chromosome. So it should be 4. And then beta thalassemia, gene is located on 11th chromosome. That means first one. Klinefelter syndrome, this is extra X chromosome present in the male, Klinefelter male. So X chromosome can be matched with this 3, 4, 1, 2. 3, 4, 1, 2. Fourth option is the correct match for that. Then 185, again a matching question. Pons. Pons connects different regions of the brain. So third one. Hypothalamus. It has got neurosecretory cells which secrete many hormones. And then medulla. Medulla has centers for involuntary activities. So controls respiration and gastric secretions. 2. Cerebellum. Cerebellum provides, because it is highly convoluted to accommodate many neurons. So provides additional space for neurons, regulates posture and balance. So cerebellum should be matched with the first one. So correct combination 3, 4, 2, 1. Third option is the correct one. Okay. Then the next question. As per ABO blood grouping system, blood group of father is B positive, mother is A positive and child is O positive. Their respective genotypes are. Child would be O group only when both the parents are at least heterozygous. Heterozygous. See, father B heterozygous, mother A group heterozygous. So, small I can be inherited one from each parent by the child, which makes the child O group. So, A is correct combination, B is not possible. In this case, father is homozygous B and mother is homozygous for A, child would be AB group. O is not possible. And third one, it is actually AB. Father is said to be B group, and this representation itself is wrong. And then again, father is B group, it is given IA, and anyway, child is O group, IA small I is not possible for the child to be O group. So that is wrong. Again, E, child is O group, AB is not possible there. So all others are wrong combinations. Only first one gives the correct data. So A only is the correct option for that question. Okay. And then again, matching question. Unicellular glandular epithelium. Unicellular glands, example given is goblet cells. So goblet cells of alimentary canal can be taken as the correct match for this. Compound epithelium, it is found in dry surfaces of skin, moist surface of buccal cavity. So fourth one. And then multicellular glandular epithelium. All except these goblet cells and few examples, most of the glands in our body are multicellular only. So salivary gland is to match with this, and endocrine. Pancreas is having both exocrine and endocrine portions. So endocrine gland epithelium can be seen in pancreas. That means 2. 3, 4, 1, 2. Fourth option is the correct combination. Then identify the correct option A, B, C, D. See, GnRH, gonadotropin releasing hormone, it activates or it causes release of two hormones, LH and FSH. So A should be FSH, and then LH acts on Leydig cells, target cell, Leydig cell, and FSH acts on Sertoli cells. So C must be Sertoli cell, and Sertoli cell secretes some factors which help in spermiogenesis. So correct option is FSH, Leydig cell, Sertoli cells, and spermatogenesis, actually spermiogenesis. So this is the correct one. Next one, 189. Gauss competitive exclusion principle states that two closely related species competing for different resources cannot exist indefinitely. This statement would be true if it is given for same resources. If they depend on different resources, there will be no competition. So members competing for the same resources, this is applicable. So this statement is wrong. And then statement 2, according to Gauss principle, during competition, inferior will be eliminated. This may be true if resources are limiting. Yes, when the resources are limiting, then the superior one can eliminate the inferior one. So this is wrong. So this is true. That means statement 1 is false and statement 2 is true. First option. Next one. This is about digestive system of cockroach. Structure used for storing of the food. Crop stores the food material. Okay. Ring of 6 to 8 blind tubules at junction of foregut and midgut. Those which are at the junction of foregut, midgut are gastric CK, second one. Ring of 100 to 150 yellow tubules, Malpighian tubules, and then the structures used for grinding the food. Grinding mill is nothing but gizzard. So that is first one. 4, 2, 3, 1. Second option is the correct one. 4, 2, 3, 1. Okay. And then again, next one, matching question. If you consider the sequence of the eras, Proterozoic is the old one. Next comes Paleozoic, then comes Mesozoic, and then Cenozoic. Okay. So lower invertebrates, Mesozoic era, you can match with birds and reptiles. Actually, Mesozoic era is described as golden age of reptiles. Okay. And Proterozoic era, this is very old one, and that can be matched with lower invertebrates. Cenozoic era is the recent one which is dominated by mammals. So you can take fourth one for that. And the last one, Paleozoic era, during which fishes and amphibians actually dominated. So second option. 3, 1, 4, 2. Yeah, first option itself is the correct one. 3, 1, 4, 2. Okay. And again, another matching question. So many matching questions are there in zoology. Now, coming to the first one, P wave. P wave is depolarization of atria. So third option, QRS complex represents depolarization of ventricles. Second one, T wave represents repolarization of ventricles, that means fourth one, and TP gap, that means after repolarization, again next depolarization starts, there is no electrical activity. So, heart muscles are electrically silent, first one. That means 3, 2, 4, 1. Third option is the correct one, 3, 2, 4, 1. Okay. And then next 193 question. Following are the statements about non-chordates and you are supposed to select the correct one. Pharynx is perforated by gill slits. Pharyngeal gill slits characteristic of chordates, not non-chordates. So, it is wrong. Notochord is absent, right? That's why they are called non-chordates. Central nervous system. Actually, CNS is dorsal in chordates, ventral in non-chordate. So, this is also wrong. Heart is dorsal if present. Yes. Chordates have ventral heart, non-chordates have dorsal heart. And post-anal tail is absent. Post-anal tail is also characteristic of chordates. That means correct combination B, D, E, fourth option. Okay. And the next question will be discussed by Botany ma'am.
Okay, question number 194 is from the enzyme part. They are asking for the sequence. What is the correct question? Regarding catalytic cycle of an enzyme action, select the correct sequence or sequential step over there. When you see substrate enzyme complex formation, okay, just write it like A. Then after that, free enzyme ready to bind with another substrate. This will be the last thing which is going to be happened. So write it a little bit away. Then release of the products. So obviously it will be before that thing. So I am writing C here. Then chemical bonds of the substrate are going to be broken or substrate broken. You have to take it after A here because it is happening after the substrate enzyme complex formation. Then substrate binding to active site. This will happen in the beginning. So you have to go with the sequence of E, A, D, C, B. So where the option is there. Second option you have to choose for question number 194.
Next question 195, again statement question. First statement, bone marrow is the main lymphoid organ where all blood cells including lymphocytes are produced. True. Lymphocytes are also type of WBC. All cells are formed in the bone marrow only. That means it's the correct statement. Second statement, both bone marrow and thymus provide microenvironments for the development and maturation of T lymphocytes. T lymphocytes are initially developed in bone marrow. Their maturation occurs in thymus. So, development occurs in the bone marrow, maturation occurs in thymus. So, this is also a correct statement. If you consider B lymphocytes, development and maturation of B lymphocytes take place completely in the bone marrow. Thymus is not involved. As we are talking about T lymphocytes, both bone marrow and thymus are related to that.
Okay, then question 196, we again have to match the list 1 with the list 2. They have given A here as a RNA polymerase 3. What it is transcribing for? It is transcribing for snRNA, small nuclear RNA, and tRNA as well. So I can match it by 4. Then is terminalization of transcription. That will be at the rho factor. That rho factor will help in the terminalization. So we can go with the 3 here. Then after that, splicing of exon. This is by SNRPs. This is slightly out of your NCERT, but you must know right now here SN SNRPs. That means for snRNA plus proteins that help to remove the exon out, right? Splicing of exon means they are making the thing, you can say from hnRNA to mRNA. And which thing helps in that splicing process? Splicing is done by RNPs, right? So I can match with the 1 here. Then Tata box. Tata box is a sequence of TA, which is having the very easy, the 2 hydrogen bonds to break that. It is towards the promoter side, right? So it can be matched by the 2. 4, 3, 1, 2. 4, 3, 1, 2. You can take it by the 1 here. 196, first option is correct.
Next question 197, again matching question. Exophthalmic goiter, it is caused due to hypersecretion of thyroid hormone and protruding eyeballs. So, Third one, acromegaly. It is due to excessive secretion of growth hormone in the adult stages. So, fourth option. And then Cushing syndrome, it is due to excess secretion of cortisol, which causes moon phase and hyperglycemia. And the last one, cretinism. Cretinism is less or hyposecretion of thyroid hormone, which causes stunted growth. So, correct combination 3, 4, 1, 2. Yep, correct. Matching is first option itself, 3, 4, 1, 2. And the next question, 198. Choose the correct statement given below regarding juxtamedullary nephrons. Juxtamedullary nephrons outnumber the cortical nephrons. Wrong. 80 to 85% are cortical. Juxtamedullary only 15 to 20%. So, this is wrong one. Second one. Juxtamedullary nephrons are located in the columns of Botany. No. These nephrons extend both cortex and medulla. Third option, renal corpuscle of juxtamedullary nephron lies in the outer portion of renal medulla. Actually, renal corpuscle of any nephron lies in the cortex only, not in the medullary region. So, that is also wrong. Last one, loop of Henle of juxtamedullary nephron runs deep into the medulla. Yes, this is the correct option. Then the next question, 199. Cerebral hemispheres are connected by nerve tract known as corpus callosum. Correct statement. Second one, brainstem consists of medulla oblongata, pons and cerebrum. This is wrong. Cerebrum is a part of the forebrain and is not involved in the formation of brainstem. Brainstem is formed by midbrain, medulla and pons. So, this statement becomes wrong due to the usage of this word cerebrum. So, first option, statement 1 is correct, statement 2 is incorrect. So, fourth option is the correct answer for that question. Last question will be discussed by Botany ma'am.
So, again they have given statement 1 and statement 2. As the statement 1, they said mitochondria and chloroplast are both double membrane bound organelle. This is right thing. And statement 2, inner membrane of mitochondria is relatively less permeable as compared to chloroplast. This somewhat a little bit tricky type of thing they asked, because they are comparing the mitochondrial inner with the chloroplast. But you know that it is less permeable mitochondrial inner membrane. Reason why? Because they are keeping the H plus toward intermembrane space. This is right. If it is more permeable, they cannot keep that difference of H plus from intermembrane space with the matrix. They can easily come back as well. But they are keeping that difference, that's because it is less permeable, right? So we can go with this statement as a right statement. So what answer we can go with? That both statement 1 and 2, that will be correct. Which option? That is the second option. Both 1 and 2 are going to be correct. So this is all about the bio part. Thanks a lot, students.
Okay, good evening everyone. I hope you did your paper well. So let us discuss chemistry paper. The first question is very easy. They're asking which one is not a redox section, right? So first of all, in a redox section, oxidation number should increase, should change, right? So they're asking not a redox section, right? Fine. You will see. So if you see in this question, here beryllium is +2 only. Fine. Okay. Sorry for disturbance. Now you can see they're asking which one is not a redox section. Fine, bacha. If you see in third option, beryllium is +2. Here also +2. Here Cl -1, Cl -1. Sodium +1, +1. So you can see none of the atom is changing its oxidation state. That's why this is not a redox reaction, right? You can see any other option like here hydrogen was 0 but become what? +1. Cl was 0 become -1. So atom are changing their oxidation state. That's why these are redox reaction and this is not a redox reaction, right? Next one is we see they're asking number of sigma and pi bonds. If I go in ethane, you can see beta, in ethane you will have here CS3, CS3, right? In ethane you will say, in ethane you have what? 2 bonds between what? Carbon, carbon. In C2, if you take this is carbon, it means they are saying C2, right? In C2 you will find 2 pi bonds. C2 will have 2 pi bonds only. Remember, it's a very important question. C2 have only 2 pi bonds by the MOT theory. Remember, special question, right? And next one is ethyne. So ethyne though you know very well, 1 is sigma and 2 are pi. Fine. So I am saying once again, in ethane only sigma bond, in ethene 1 sigma 1 pi. In C2 you can prove it by MOT theory, in which you have both the pi bonds in C2 using MOT. In ethyne 1 sigma and 2 pi, right? From here you can easily find, like I can say ethane they are saying, so only 1 sigma bond, right? 1 sigma 2 pi they are saying, 2 sigma 2 pi they are saying, 1 sigma molecule and 1 sigma. Okay, I think some mistake is there. So ethane only 1 sigma, ethene 1 sigma, 1 pi, carbon 2, it's C2 actually, both pi. Ethyne means what? 1 sigma 2 pi, right? Okay. Next question is the most stable carbocation they're asking. The most stable carbocation, right? In this case, if you see, I am having 3 alpha hydrogens here, 2 alpha hydrogens here, right? And 2 here. So here we have 7 alpha hydrogens. Fine. 7 alpha hydrogens, making it very stable. Here you have only how many? 1 alpha hydrogen. Here how many? 3 and 2, 5. And here how many you can see? 1 here and 2 here. So because of the most stable, why it is most stable? Because of a lot of hyper conjugation structure. That's why it is most stable carbocation among these, right? Okay. Next one they're asking, these complexes will show which type of isomerism, right? Here very easy, NO2 is a ambidentate ligand, right? In ambidentate ligand you can connect metal with the 2 different atoms in the ligand, right? Like in NO2 ONO or NO2 directly. So this will show which one? Linkage isomerism, right? This Br can go inside, SO4 can come outside. It can show which one? Ionization isomerism, right? Next, if you see here, if I will put cobalt here and chromium here, so coordination between the metal and the ligands will change. So that's why it can show which one? Coordination isomerism. And here, if I keep 1 water molecule outside, keep 5 water molecule inside with 1 Cl. So when number of water molecules acting as a ligand are changing, what we call them? We call them solvate isomerism, right? So you can easily check your answer from here. Fine, bacha? Clear? Okay. Next one, they're asking the electronegativity order. Definitely, the most electronegative elements are three: fluorine, then oxygen, then nitrogen. Very easy. You can check here. We have fluorine maximum, then oxygen, then which one? Nitrogen. Clear, bacha? And if you go down the group, carbon, silicon, germanium, right? So we know when you move down the group, electronegativity decrease, right? That's why from that point easily you can say fluorine, oxygen, nitrogen, then carbon and silicon, the right order for electronegativity. Fine. Next one, again very easy question, right? What we have here? Ammonia. Ammonia we know very well. Ammonia have one lone pair and three bond pair. So what is it? Trigonal pyramid. Easily you can see, it means A is getting which one? 1. Only one option have what? A1. You can directly get your answer, right? Still you can say BRF5 is which one, beta? It is square pyramidal, right? XCF4 is square planar. 4 bonds, one lone pair upside, one lone pair downside, right? It is square planar. SF6, the most easiest one, which one? Octahedral. Fine. You have to save your time by this technique. If you are getting one, you can go for that directly. Okay, fine. Okay. Next one, if you see, which does not show the -2 oxidation state. Okay, -2 mainly is shown by the known metals, right? But when we move down the group in oxygen family, oxygen, sulfur, selenium, tellurium, polonium, so metallic nature increasing. That's why the polonium do not show the -2, or I will say very less chances are there. Okay. So -2 tendency decrease down the group if you move down the group. Fine? Clear? Okay. Next one, they're asking the graph between ln k versus 1 by t. So I will say from Arrhenius equation, if you take ln, so you'll get ln k, ln a here, and minus of ea by rt, right? I can say 1 by t. So this is our what? X. It's our y. It's our c. So I can say y equals to, this is our c, and here it is what? Mx. So slope is negative, minus ea by r. Negative slope means the line should come down, the most easiest one. Clear, bacha? Line is coming down, right? ln k versus 1 by t. Fine, bacha? Okay. Next one, the compound that will undergo SN1. Fine. A thing which can make a very stable carbocation will have more chances for SN1 reaction. Fine. So if you see, which of these is making the most stable carbocation, right? You just break Br from here, positive I'm getting, very unstable chances. Here if you remove Br, right, I'm getting positive charge carbocation. It is very stable because of 3 alpha hydrogens and benzene here. Lot of conjugation, lot of hyperconjugation. That's why its chances for SN1 will increase, right? Clear, bacha? So that's why you will say it has the most probable chances for SN1, right? Okay, right. Oh, just wait for 1 second. Let us rectify this issue. sorry for the inconvenience. I think some issue was there in that PDF. Let's come back to our main part again, right? We are having this PDF. So please look at this. First of all, they're saying they have given the reaction and they're asking, they have given the concentration of reactants and products as well, right? So they're asking a reaction will go forward or backward. So we can find Q here, right? How we can find Q, the reaction quotient? They're saying BC amount is given, right? They are giving the B concentration, they are giving the C concentration divided by what? They are giving A concentration square. If you see carefully, B, C and A all are same given. So if you put this concentration here, here and here, as they are same, so Q is coming 1. If Q will come 1, 1 will be what? 1 will be more than this, more than the Kc value, right? You know, if Q is coming more than Kc, then reaction go where? Backward to attain equilibrium, right? So that's why we'll show reaction has tendency to go backward in direction. Okay, Q is more than Kc. Fine, bacha, right? The next thing they are saying, the highest number of helium atoms is in. You have to find number of moles, then what? Number of atoms. So if you see, in first case, you can apply the formula given volume divided by 22.4, 22.7, fine. So from here you will see almost you are getting how many moles? Only 0.1 mole. Here I am having 4 moles. Here I am having only 1 atom because it is only what? 4 U of helium, very less amount, right? And 4 gram helium is 1 mole again. So in this case, we are having the maximum number of atoms. 4 moles will have maximum number of atoms, right, bacha? Next one they are saying a compound with formula C6H14 has 2 tertiary carbons, right? The IUPAC name they are saying. If you make a compound with 2 tertiary carbons and with only 6 carbons, right? It is the only possibility. You can see tertiary carbon, tertiary carbon. Clear, bacha? That's why you can say very easily. It's your answer, right? What is the name of this? It is 2,3-dimethyl butane. Fine, bacha? Very good. Next thing they are saying, if I convert H2O to O2, so O is going from -2 to 0. -2 to 0, how many mole electrons required? 2 mole electrons, means how much charge required? 2 Faraday, right? Mn plus 5 to going to Mn plus 2, right? How much? Mn plus 7. Mn plus 7 to Mn plus 2. So how much Faraday required? 5 Faraday, right? So for this I should get which one? 5 Faraday. Here 1.5 they are saying calcium, right? Calcium 2 they are saying calcium 1.5 mole. So be careful. For 1 mole calcium 2 Faraday. For 1.5 mole how much Faraday? 3 Faraday, right? For this you can get easily the 3 Faraday. And 1 mole FeO. Fe is what here? Fe is plus 2. And here going into Fe plus 3. So how much Faraday required? 1 Faraday from here. Okay, easily you can get your answer from here. I think for A you are getting what? For A we are getting 2, right? That's it. And B we are getting 4, right? And C getting 1, 5, right? So second is the right answer. Fine, bacha? Easy. Next one. Okay, fine. Now they're asking the electronegativity order. I just few minutes back I told you the order, which one? Fluorine, nitrogen, oxygen and silicon. This I also explained the graph of ln k with 1 by t, negative slope, right? So what is coming here? First one, very easy. Now they are saying Kp Kc is not equal. The relation is Kp equals to Kc RT delta Ng, right? If delta Ng will be 0, then I can say Kp equals to Kc. But if delta Ng is not 0, then I will say Kp is not equals to Kc. You see, in this case 2 moles reactant side, 2 mole product side. But in second option, 1 mole reactant side and 2 moles where? Product side. So delta Ng is how much? 1. That's why you will say it will, in this case, delta Ng is not 0. So Kp Kc is also not equal here. Fine. The next one I told you, the most stable carbocation among the following. Fine. I told you just now it is having 7 alpha hydrogens. Lot of alpha hydrogens make it most stable carbocation out of these. Getting my point? Fine. You can check the alpha hydrogens because none of these is having conjugation. So only alpha hydrogens will decide here the stability. Fine, bacha? The next one is activation energy can be found how? We know this formula, very common formula, beta. If you see that log k2 by k1, right? What comes here? EA divided by 2.303, right? Into what here? Into what? I will say 1 by T1 into 1 by T2. Fine. So what is coming here? 1 by T1 minus 1 by T2. So if you know the rate constant at two different temperatures, like here, I know at T1 it's K1, right? And at T2 it's K2. So I can find what? Activation energy just from NCERT, the most simplest, right? So we can find the rate constant. Definitely, if you know the rate constant at two different temperatures, using this we can find activation energy. Fine, bacha? Okay. Next one. Given is the order for the boiling point in group 16 hydrides. As mass will increase, the van der Waals force will increase, size increase, right? And that's why, because of more van der Waals forces, the boiling point increase when we move down the group, except water, because water can make what? Hydrogen bond with each other. That's why its boiling point and melting point have little exception. Clear? So you will see here, yes, order is right. Why S2 is exception? Because of hydrogen bonding. Okay, fine. Next one, they are saying on the base of molecular mass, S2 is expected to have lower, as I told, because mass is less, size is less. But what will happen here? The thing is, hydrogen bonding is making it exception, right, bacha? That's why both are right statements. So we'll say, okay, statement 1 and 2 both are true. Fine. The next one, if you say, which one of the following alcohol will give Lucas test instantaneously? We know tertiary, what is the reactivity order? Tertiary, secondary, primary. Tertiary give instant turbidity with Lucas reagent, right? So we can say here, we are having tertiary alcohol here. Fine. Now, the next one, if I go, okay, aniline does not undergo Fieldcraft alkylation. Why? Because beta, it's very important. You have seen in NCERT also. Because here aniline react with the Lewis acid AlCl3, which is the catalyst for Fieldcraft reaction. If catalyst will not be available, then how it will give reaction? So because of reaction between the catalyst part of Fieldcraft reaction and aniline, this will not give Fieldcraft reaction. Very good. Aniline cannot be prepared through Gabriel phthalimide. From Gabriel phthalimide, we can make only, which one? Only primary aliphatic amines. Getting my point? Why? Because if in the Gabriel phthalimide, if you will take aryl halide, then what will happen? We know that aryl halides are very less reactive for nucleophilic substitution reactions, right? Because what they have here? Partial double bond nature. Okay, so you can't take this one. Fine. Okay, that's why. So you are right. You cannot take which one? You cannot take here. You cannot make here which one? Aniline. You can make only aliphatic amines, right? Next one is, okay, they are saying ammonia. Just now we did ammonia is trigonal pyramidal, right? BRF5 is what? Square pyramidal. XCF4 square planar. SF6 octahedral, right? So from here easily we can find A is 1. Only one option is having A is 1, right? The next one you can see ML. ML is which one? ML is you can say your magnetic quantum number. It will tell you the orientation of what? Orbitals. So you can go for which one? Third one. Orientation of orbitals. Fine. If you go for MS, MS will tell you what? Orientation of electrons. L will tell you what? Shape of orbital. L0, 1, 2, 3 will tell you it is S, P, or D, or F. That's how you can find what is the shape of these orbitals. Okay. Now N will tell you the size of orbital. Okay, means if N is increasing, size of orbital will increase. Fine. From here you can find the option. Clear, bacha? So this is giving which one? This is giving us orientation of orbital. So I'm getting M as 3, right? M as 3, fine, we can check. Then this is giving B4, right? And then which one? C is giving us 1, shape of orbital, right? So you can go for third one, fine. Okay. And the next one you can say, why manganese is highly interested in coming from Mn plus 3 to Mn plus 2, because Mn plus 2 is very stable. Mn plus 2 is what? D5. Mn plus 3 was what? D4. So if it go here, it will get stability, and that's why it is very much interested in this reduction, and that's why you will say it is having high positive value of this reduction potential. Fine. Okay, so now some questions analysts will discuss, right?
All right. Very good evening to all of you. I hope you have written your paper well. Okay. And all the questions which you have seen in the chemistry are, most of the questions are from the NCERT and whatever you have written notes over here in the classes. So that's good thing. Okay. Let us start with question number 66. Entropy increases in which of the following processes? Delta S positive. Whenever we go from liquids to solids to liquids, or liquids to gas, in that case we know it increases. A liquid evaporates to vapor, yes, delta S will be positive, all of you know. Temperature is lowered, no. When you decrease the temperature, we know entropy is inversely proportional to temperature, so it's not correct. I heat a solid and a gas is coming out, so obviously it is also increase in entropy. 1 mole of gas giving you 2 mole of gas. So whenever delta Ng is positive, delta S is also positive. So this is also correct. A, C and D all are correct here. Fourth option. Okay. Next one. Energy of electron in the ground state of helium plus is given -6. These type of questions many times you have done in your class. How do we do them? We do them by taking the ratio. You say that I will take the atomic energy. So energy of electron in helium plus by energy of electron in beryllium plus 2 plus 3. Okay. Beryllium plus 3. Energy formula is what? Energy is directly proportional to Z square inversely to N square. So Z of helium, N of helium by Z of beryllium, N of beryllium. Now we'll put the values. This value is given -x. This one we have to find. For helium, Z value is 2. So 2 ka square, which is 4. Shell first is given. So 1 square I'm writing. Then Z of beryllium, which is 4, 4 square. And the second shell they are talking about. 2 square, 16. 4 square, also 16 will cancel out. So energy of beryllium will also come -x. Okay. Simple question. All of you have done correct it or not, please. Tell in the comment box how many people have done this one correct. Option 2. Quickly tell. Okay. If you have done correct, just write correct. 68 already it is discussed, which is not a redox reaction. Neutralizations, precipitations, and some decomposition like decomposition of calcium carbonate, magnesium carbonate are not redox reaction. All right. Next one. Next question is, we have to reach from this particular alkene to, we have to oxidize this corner carbon. We have to oxidize this corner carbon. How to oxidize the corner carbon to aldehyde? I can first convert it to alcohol. If I can first convert it to alcohol, for this you need anti-Markovnikov addition of water. Anti-Markovnikov. You want OH negative to go here and H positive to go here. So it has to be anti-Markovnikov addition. For that, what do? I can do hydroboration oxidation reaction, which is B2H6 H2O2. Okay. So first step will be this. Then second step, you could do PCC PDC, because you want to convert a primary alcohol to aldehyde. PDC, PCC, CrO3, anything will work here. Okay. So here the best option looks like H2O, no. H2O, no. BH3, yes. PCC, yes. Third option. Okay. Third option is correct here. Next one. On heating, solid directly changes to vapor, means we are talking about here sublimation. Okay. This is the principle behind sublimation. Next one. This one sir has already discussed with you. NO2 being an ambidentate ligand will show linkage isomerism. It is an anion outside, an anion inside. I can exchange both. I can get the ionization isomerism. And whenever two brackets are there, coordination isomerism. Whenever water is there, solvate isomerism will be possible. So accordingly, you can check the answer 2, 3, 4, 1. 2, 3, 4, 1. Option 2. Option 2. 2, 3, 4, 1. Okay. Next one. 72. Two statements are given. Let us see. Both CO ammonia 6 plus 3 and CO F6 minus 3. So in both of them, cobalt is present in plus 3 oxidation state, which is 3d6 configuration. 3d6 configuration. So when you take cobalt 3d6 with strong ligand like ammonia, there will be a lot of energy gap between T2G and EG because it's a strong ligand always create more gap, and whereas a weak ligand will create a little bit gap only. So in the case of F negative as ligand, we have to follow this particular diagram. So in the case of ammonia, there will be forceful pairing taking place. 1, 2, 3, 4, 5, 6. It is diamagnetic. Whereas here 1, 2, 3, then no forceful pairing. 4, 5, 6. So it is paramagnetic. Okay. Now question becomes very simple. They are octahedral but differ in their magnetic behavior. Absolutely correct. This is dia, this is para. Absolutely correct. Both the statements are true. Second option goes from here. Okay. Next, what is Fehling solution A? Fehling is made up of A and B. Fehling A is nothing but aqueous copper sulfate. This is Fehling B. Okay, alkaline solution of sodium tartarate. Okay, sodium citrate and sodium tartrate. They are there present in the Fehling B and also present in the Benedict solution. So Fehling A, they are asking aqueous copper sulfate. Next one, intramolecular hydrogen bonding. By chance, 2 days back only when we were discussing how to get better marks in chemistry, exact same example we discussed. Intramolecular means orthonitrophenol. One of the most favorite example of all the teachers is orthonitrophenol, option 2, orthonitrophenol. Okay. Don't go by whatever is marked here. It is someone's paper who might have done many mistakes. So you go by what I am telling. Okay, second option, which is orthonitrophenol, intramolecular hydrogen bonding, and that is why it is steam volatile. Okay. Next one. Pentane, isopentane, neopentane. This is pentane, very high surface area. That is why very high boiling point. This is isopentane, little less surface area. This is neopentane. I can complete the neopentane in very small region, whereas isopentane takes a lot of surface area. So boiling point will be in this order. Now what they are saying, boiling point is in this order. That's correct. When branching increases, molecule attains shape of a sphere. Just now how I have made. This is looking like a long molecule. This is looking like a circular molecule. Thereby intermolecular forces become weak, and it's absolutely correct. They become weak. That's why the boiling point decreases with branching. So both the statements are actually true here. So statement both 1 and 2 are absolutely correct. Second option. Next one. Among group 16 elements, among group 16 which does not show -2, this one just now Amit sir had discussed. Polonium, being most metallic, will have least tendency to show negative oxidation state. Oxygen, sulfur, selenium, tellurium, polonium. Polonium being the worst in the case of negative oxidation state. Next one, Henry's law constant is given. Many people have done this question wrong. I don't know why. We know that KH is inversely proportional to solubility of the gas. Now, what is the order of KH here? You can see here KH of A is given 145, which is very, very high. Then KH of C is given 35. Then KH of B is given as 10 power -5. So obviously solubility will be B the maximum, then C, then A. BCA. It's a simple question, very logical question. BCA. BCA, option 3. Okay. Moving to the next one. 78. 1 gram of NaOH is taken. It is treated with 25 ml of 0.75 molar HCl. How much mass of NaOH is left unreacted? Okay, so this one is a good question. Just a question related to mole concept. First of all, you think about the reaction. Okay, reaction is NaOH reacts with HCl, gives you salt plus H2. NaOH is given 1 gram. Okay. HCl they have given molarity and volume. So if I say 1 mole NaOH reacts with 1 mole HCl, if I know the moles of HCl, so moles of HCl will be nothing but what? Molarity into volume. Volume must be in liter. And the molarity is given 0.25. Volume is given how much? Molarity is given 0.75. And molarity is given this one, and volume is given 25 ml. Divide by 1000 I'll do. So these are the moles of HCl. Now, as they have said, calculate the mass of NaOH unreacted. Means NaOH is your excess reagent and HCl is your limiting reagent. So if I know the moles of HCl, same must be the moles of NaOH reacted. So moles of NaOH must also be equal to moles of HCl. What will be the mass of NaOH reacted? Mass of NaOH reacted, it will be moles of NaOH reacted into 40 gram. Into 40 gram. So we get mass of NaOH reacted, mass of NaOH finished, I can say, is how much? Moles, which is 0.75, I will write it as 3 over 4, 25 by 1000 into 40 gram. Into 40 gram. So after solving it, we'll see how much we get. 4 and 40 cancels out. 10 times 0, 0. 25 4s are. So I'm getting 3 over 4, which is 0.75 grams. 0.75 grams is not the answer. This much has reacted. We started with 1 gram, 0.75 reacted. How much remaining? 0.25. 0.2. Ashley talks. This is the mistake what everyone has done. Beta, it's not only 35 bar. Everyone is kilobar, no. Everyone is kilobar. Where did they mention 145 is bar? Where did they mention 2 into 10 power minus 5 bar? They are saying 145, 10 power minus 5 and 35 kilobar respectively. Means all are in kilobar. Where did they mention 2 are in bar and 1 in kilobar? All are in kilobar only. Are you getting the point? Yes or no? That's why students have done mistake here. Read question carefully. Respectively means all are in the same units. Okay. Our masses are 2, 10 and 15 kg respectively. So all are in kg only, no. First 2 and 10 are not in grams. When I say our masses are 2, 10, 50 kg, it's all 2 kg, 10 kg, 50 kg. Okay. So 0.75 has reacted. Remaining is 0.25. Third option. 250 milligrams. Okay. Next, spin only magnetic moment is same for what? Titanium plus 3. Titanium plus 3, I hope all of you know the configurations of titanium. Scandium, titanium, it will be D1. Chromium plus 2, D4. Manganese plus 2, D5. Iron plus 2, D6. Scandium plus 3, D0. Okay, so unpaired electrons are 1 in titanium, 5 in manganese, 0 in scandium. So ACE, none of them is equal. D4 means 4 unpaired. D6 also means 4 unpaired. So B and D both are correct here. Both are same. B and D both are same here. Okay. All right. I'll discuss this one and then Amit sir will discuss the next few. Next one is we have to match the following. First one is a very typical example of reductive ozonolysis. I have to give 1 oxygen here, 1 oxygen here. So it is giving us 2 moles of ketone. A matches with 4. A matches with 4, A matches with 4. So other options I'll check. In second and fourth, we are having the 50-50 choice. B1, anyways we won't check. C3, C2, we'll check for C3 only. C is a alcohol, a secondary alcohol going to what? Going to a ketone. So I can do its oxidation. Secondary alcohol to ketone, oxidation we have to do. And for the last one, now this oxidation can be done by KMNO4 as well. It can be done by CrO3 as well. Okay. But for this particular case, Nivedita, same problem for you. Nivedita, 35 kilobar is not only 35 kilobar. Others are also kilobar. If they haven't mentioned the units, only once they have mentioned the units. So all of them are kilobar. Where is it written 145 bar? Everywhere it is kilobar. Okay. 145, 10 power minus 5, 35 kilobars respectively. All of them are kilobars. Okay. So, I was talking about this secondary alcohol to ketone. I can go by CrO3 also. If it was given CrO3 H2SO4, it would have been a better option like Jones reagent. Since just CrO3 is given, we'll take it as aqueous CrO3. And obviously, when you have alkylated benzene rings with alpha hydrogen, they can be oxidized to benzoic acid by alkaline KMNO4. This reaction to all of you know. Okay. So, D will match with the third option. C will match with 2 and 3 both, but we'll go for the third option being there is no other one. So C with 2, D with 3, C with 2, D with 3, option 4. Okay, because D cannot match to anyone else. Okay, all right. Next 10 questions or more, Amit sir will just take.
Okay, fine. So now we'll see 81 question beta, right? Okay, we have done this already. You know very well ethane is having a carbon which bond, single bond. Okay, so CEC single bond is where only one sigma bond, right? So for first I'll go for 3. Ethene is having double bond in carbon, one is sigma and one is what, pi, right? So for ethene we'll go for which one, 1 sigma and 1 pi. So you can see for A I'm getting 3, for A I'm getting 3, okay, 2 options are there, and for B I'm getting 4, 2 options are there. Okay, A3, B4. Let us go for next one. C2. As I told, by the MOT theory, you will find it is the most common question which tells us C2 is having both pi bonds, right? So, you will go for which one? 2 pi bonds, right? So, for C, what is coming? 2 is coming. C2, right? This is given answer. Still, if you want to go for ethine is having 3 bonds. Out of 3, the middle one is which one? Sigma, and 2 are pi. So, yes, your answer which one? Fourth one, right? Next one, if you go, the compound that will undergo SN1 reaction with fastest rate, right? So carbocation formed will be stable, then the reaction will be very fast, right? So in this case, you can see, if I remove Br, I am getting a carbocation with 3 alpha hydrogens as well as a lot of conjugation, right? So yes, this is the highly stable carbocation. This is having no conjugation, highly unstable. If positive charge is present on what? Benzene, highly unstable, right? Here also I am getting secondary carbocation, nothing special, right? The first one is most reactive for the SN1 mechanism, right? Okay, the next one, if you go, they are asking the ionization enthalpy order, right? So, bacha, we know it very well. Then when you go in a period like lithium is there, beryllium, boron, carbon, nitrogen. So, in general, the ionization energy increase, that's fine, okay, but the reason here, the boron is having configuration NS2, NP1, and beryllium have how much? NS2. Because of penetration power, the S electron is highly, or you can say strongly attracted to the nucleus, whereas in boron it is less attracted with the nucleus or held by the nucleus, right? That's why you can say beryllium is having more than what? Boron. Or little bit you can say its configuration little better than this one, okay? Main reason is penetration power. S electron have more penetration power than which one? P1. Very important exception, right? So we can see nitrogen, okay, carbon, boron, but beryllium is more, okay, right? So nitrogen very high, carbon, beryllium is more than what? Boron. That is the only exception here. Fine, bacha? Good. Next, they are asking the reagent with which glucose does not react to give the corresponding test or products. Clear, bacha? First of all, if you see the topic in NCERT, limitations of the open chain structure, they have clearly mentioned. We cannot take glucose only as open structure and it is having aldehyde. When it will make cyclic structure, it is having hemiacetal group. So we have to see from both the ways, right? And experimentally, it is definitely given in NCERT. It do not react with what? With NaHSO3, right? This is the limitation of open chain structure. Clear, bacha? So this is definitely given in NCERT, and with SCIF reagent also it don't react. So you can see only one option have what? E, that is fourth option, B and A, right? E, it is given in NCERT that it don't react with SCIF reagent and NaHSO3, right? Clear. The next one is they're asking the isothermal process. Isothermal means what? Delta T0, there is no change of temperature. If you go for the isochoric, that means delta V0, no change in volume. They are saying which one now? Isobaric, no change of pressure, pressure is constant, and adiabatic means what? Q is 0, no transfer of heat, right? From here you can easily find, right? Isothermal means what I will say? I will say carried out at constant temperature, right? And you see for A, only in one option 2 is given. So you can easily go. You can get your answer just in few seconds very easily. Right? Clear? Next, if we go, you can check beta here. B means 3. They are saying B means isobaric, means what is constant? I will say they are saying B3 means what? Isobaric means pressure is constant, right? So where they have given, yes, this is the pressure constant, right? Isochoric, sorry, it is isochoric. So at constant volume, isobaric is what? Constant pressure. Adiabatic, which one? No heat change, right? Easily you can get your answer. Fine, the most easy. The next one, if you go again from practical chemistry, when we want to make Mohr salt, we mix our ferrous sulfate and ammonium sulfate in equimolar ratio. As you are taking FeS, Fe plus 2, it can be oxidized to Fe plus 3 also. To stop this oxidation, we put 2-3 ml amount of dilute sulfuric acid, right? The very easy question once again. So you can say dilute sulfuric acid we are adding just to stop the oxidation of Fe plus 2 into Fe plus 3. Fine. And you will see it will not hydrolyze to stop the hydrolysis, right? What they are writing here clearly, they are saying which of the following acid is added to prevent hydrolysis, right? So we will use dilute sulfuric acid to avoid the hydrolysis of Fe plus 2 ion here. Fine. The next thing you will see, identify the correct answer. CO3 2 negative, right? This is our CO3 2 negative, right? Now in this structure, this carbon having double bond, maybe this carbon can have double bond now, and next possibility is there, this carbon is having double bond. So you can see here we are having 3 resonating structures, or you can say canonical structures of carbonate ion, and fourth will be the resonance hybrid, right? You will not count it in resonating structure. So you can say clearly how many we are having? We are having 3 canonical structures of carbonate ion, right? For ozone, if you check, for ozone there are only 2 resonating structures. So that definitely it is wrong. They are saying 3 here. BF3 have a symmetrical structure, so 0 dipole moment, right? And the next one is NF3 is greater than NH3. No, very important point from NCERT. Because of proper directions, in NF3 some cancellation will happen and net dipole moment become what? Less. Like in lone pair, in NF3 is pulling upside but F is pulling downside, so little cancellation is there. Whereas in NH3 cancellation is very less, so net dipole moment of NH3 is more than what? NF3. Fine, right? So very easy to find the answer, right? Now we can come to the 88. Here they are saying to find the empirical formula. Last year also they asked a very easy question from here. We can see one. They are saying we are having 3 elements. I am saying A element, B element, and which one? C. They have given their percentage like 32% of A. Weight is given 32 and B is given 20 and remaining is how much? 48. This is for C. You can imagine we are taking 100 gram of this compound. So in that you can say 32 gram A, 20 gram B and 48 gram what? C. Now I want to find the molar ratio. Their moles are, molar weight is given. So how we can find 32? What is the molar weight for A? They are saying, okay, it is giving 64. Here how much given? 20 by 40, right? And here it is 40, 8% coming, okay, 48 and divide by how? 32. Fine. So if you solve this thing, what is coming from here? 1 by 2, 1 by 2, and what is coming? 3 by 2. Fine. So if you find the ratio of ABC here, what is coming? ABC molar ratio, which are how much coming from here? 1, 1, 3. Very easy to find, right? So 1, 1, 3 I can say, okay, it is here only A1, B1, C3. Fine, bacha. Next thing is 89. If you check carefully, they're asking, given below are the certain cations using inorganic qualitative analysis, arrange them in increasing order of group number, right? Just again from practical chemistry, if you go, copper plus 2 is from which one? Second group, third, fourth, fifth and sixth, right? Clear? So if you go in the increasing order, B, A, D, C, you can find here B, A, D, C, yes. I'm saying once again, B, A, D, C, then what is remaining? The last one is E, okay, right? From practical chemistry, fine. The next one, if you see, the work done during the reversible isothermal expansion of 1 mole of hydrogen gas from pressure 20 to 10 atmosphere, right? 20 atm to 10 atmosphere, right? First of all, you can give your answer in 1 second. Expansion is there. Just they have mentioned work done and they are saying expansion. So it's very easy to find. If you see, only one is negative given, right? Expansion means negative work. When they don't mention anything, work done by gas or work done by atmosphere, no. So you can apply the formula. Work is what? W equals to minus of 2.303, right? You can say nRT. In terms of volume, we say V2 by V1, right? But it's pressure, right? So we know that if temperature is constant, P1 V1 equals to what? P2 V2. So from here we can say V2 by V1 coming how much, bacha? P1 by P2. So just put the value of P1 by P2 here and calculate using the formula. You will get your answer. Easily I am saying, just see the negative sign and you can get your answer. Fine, bacha? If any questions, others will discuss. Okay.
All right, students. We'll go for the 91 now. The plot of osmotic pressure versus concentration is given. So what is osmotic pressure? It is CRT, as all of you know. ICRT, but because it's not given that it's a electrolyte or non-electrolyte, I'll just take CRT, I value as 1. Pi versus C. This is our y-axis, this is our x-axis. Y equal to mx. So this becomes the slope RT. So this slope is RT here and the slope is also given here. So they are saying slope is 25.73 liter bar mole inverse. So value of R into T is given 25.73. Whenever they give the value of R, then in that case you take the value of R exactly. If you take 1 by 12, you might do some mistake in the calculation. Okay, so 0.083 exactly I'll take. 0.083 into temperature equal to 25.73. From here we find the value of T, and I have done it myself. It is coming 310, but bache, it is Kelvin. It is all coming in the SI unit, Kelvin. So it is 310 Kelvin. Please don't put the answer 310 degrees Celsius. Many students have done this mistake. Don't put this. No, this is not the answer. 310 Kelvin means how much temperature? So it means it is 37 degrees Celsius. So option 2 will be there. Option 2 is correct. 37 degrees Celsius. Okay. Raghavendra, you did that. Huh? You're telling like this. Next one. Major product A and B in the following reaction are. First reaction is our SN2 reaction. PBr3 will do nucleophilic substitution of Br negative, will substitute the OH negative. So we know that here a Br will come. So option A, I mean the product A has to be this bromine substituting the OH. Next time I'm adding alcoholic KOH, which will do beta elimination to give the ZZ product as the major product. ZZ product as the major product. So more substituted alkene will be formed from here. So this is our first product A. Now this beta hydrogen will be removed by alcoholic KOH. I hope all of you know this, and it will go by ZZ rule. This will be the product. Okay. 1-methyl cyclohexene. B is also this. So second option comes from here. Second option comes from here. You would have done mistake if you have done mistake. You would have done it between second and third option. Third option is not correct here. Third option would have been, no, third option is not possible only. If this pi bond was present here, let's say, then it would happen with a bulky base like tertiary butoxide. Okay, next one. Mass is given, so mass in grams of copper deposited. Okay, so it's a Faraday's first law question. All of you know the Faraday's first law. What is the formula of Faraday's first law? It is weight of product, means copper, equal to equivalent mass of copper E by FIT. Faraday's law of electrolysis, E by FIT. So I'll find the weight of copper. Equivalent weight means molar mass 63 gram by N factor, which is 2 here. I, which is given 9.64 something, and time is 100 second, and at bottom Faraday 96,500. So exactly if I check, it will cut at 1, 2, 3, 4, 10,000. It will exactly cut at 10,000. 9 times 10,000 is 90,000, 96,000, 100, 2 zeros. So I get 31.5 by 100, which is 0.315 grams. 0.315 grams. Okay. Again, a question which is there in all of your notes, exactly same values would have also been there if you check. Okay. Next one, 94. This one is a little longer question, which is the only long question in today's paper. Okay. All of you can see here and check. Although now those who are becoming doctor this year, those who are getting 160, 170 marks, you might skip this question now. You don't have to do any more chemistry. But those who are planning to do next year as well, you can see here, maybe next year it can come in section 1. Okay, the question goes like this. They are saying there is a reaction, 2NO gives 1N2 plus 1O2. They are saying consider the following reaction in a sealed vessel at equilibrium. So this reaction is at equilibrium right now, and its equilibrium concentrations are given to you, which are given to you as 3 into 10 power minus 3 at equilibrium, 4.2 10 power minus 3, 2.8 10 power minus 3. This is at equilibrium. That means this, why did they give this value? They only gave these values so that you can find the equilibrium constant. So equilibrium constant comes out how much? You can solve 3 into 10 power minus 3, which is concentration of N2, concentration of O2 by square of NO. 2.8 into 2.8, 10 power minus 3 also 2 times I am writing. Cancelling it out, 10 power minus 3, 10 power minus 3, 10 power minus 3 will cancel out. From here, when you solve it, okay, this value will come 1.60. Okay, you can do the solution. It's a simple one. So you will get 1.60. This is our equilibrium constant. All right. Then they started with 0.1 mole per liter NO. I'm writing this reaction again. Now I know the Kc of this particular reaction. I will start with saying the Rice method. Reaction initial 0.100. We know whenever we want to find the alpha, out of C always C alpha breaks. So if it is C, it from here 0.1 alpha will break. When 2 NO2 breaks, 1 NO2 forms. If 1 NO2 will break, half will only form. So if it is breaking C alpha, it will form C alpha by 2. 0.1 alpha by 2. This will also form 0.1 alpha by 2. Okay. I broke C alpha. I formed C alpha by 2, C alpha by 2. So remaining is 0.1 into 1 minus alpha. Remaining is 0.1 alpha by 2. Remaining is 0.1 alpha by 2. Okay. After this, now we need to just substitute this in the equilibrium constant once again. So you can put here this 1.60. Again, I am putting the formula of equilibrium constant, which will be 1.60 equal to N2 concentration to the power 1, 0.1 alpha by 2, 0.1 alpha by 2 by square of this 0.1 square 1 minus alpha whole square. Okay. So this becomes 0.1 square, 0.1, 0.1, everything is cancelling out. This will become alpha square by 4. This 2 into 2, 4 I am bringing here. So it will become 4 times 1.6 equal to alpha square by 1 minus alpha whole square. This whole thing I can write it as what? Alpha by 1 minus alpha whole square. Square will come this side, becomes root. So I get 6.4 under root 6.4 under root equal to alpha by 1 minus alpha. Now you just need to do the cross multiplication and solve for it. From here you will get the value of alpha as 0.717 exactly. A little bit of a long question. This is the only question which came as a lengthy one. I hope you have skipped this question. This question had to be skipped. It was just given so that if in case you are not able to solve any other question, you should have tried. Otherwise, it's a pretty long question. There is no other way to do in a small method. This is the only method that you can go by. I hope it is clear to everyone. You can rewind the video if you want to check the things again. Okay, next one. All right, I have a propyl iodide NaCN will make propyl cyanide, or I can say butane nitrile. Butane nitrile hydrolysis, partial hydrolysis means just one water I will add. For that, one oxygen will go here, two hydrogens will go to nitrogen. So I'll get what? Amide, butane amide. And then finally we are doing what? We are doing Hofmann bromide degradation reaction in which CO will be removed. So I get CCCNH2 propylamine. Okay, option 2, propylamine. Okay, I hope you have not done the test the way this student has done. I didn't find any option correct till now. Okay, next one. Pair of lanthanides which are diamagnetic. You can check all of their configurations and see. Right now, I'll just say you can look at cerium plus 4. Cerium, which is going to be 4F 5D 6S. 6S2 5D1 4F1. Okay, 6S2 5D1 4F1. So when I remove all the 4, it will become noble gas configuration. So obviously it will be diamagnetic. Okay, and ytterbium. Ytterbium is the second last lanthanoid. Last lanthanoid being lutetium. So this one will be 4F completely filled 14. 5D is 0, 6S is 2. Okay, 6S is 2. So when I remove 2 electrons from here, again it becomes 4F14. So this is also going to be diamagnetic. Okay, so both are diamagnetic. You can see their electronic configuration and decide. Next one. Another question which came from your notes itself. In the presence of KMnO4H plus, oxidative cleavage. Oxidative cleavage whenever you are doing and carbon is having one hydrogen, then in that case just break the carbon-carbon bond. Both sides give 2 oxygen. 2 oxygens here, 2 oxygens here. That means it will become a carboxylic acid. Cyclohexane carboxylic acid, which is option 3. Both will become cyclohexane carboxylic acid. I hope you have not made the second option. Second option will be formed if ozone ZnH2O was there. Okay, ozone ZnH2O was there. All right. Last 4. 97, 98. Last 3 are there. 98. Given below are the 2 statements. This is a homoleptic ligand. Yes, all the ligands are same. So it's a homoleptic complex. This one is a heteroleptic complex because 2 different ligands are present. It has only one kind of ligand, 2 like kind of ligand. Absolutely correct. So both are correct statements. It's a simple question. Next one, rate of reaction. So Arrhenius equation question. I'll just write the formula here for you. All of you know the formula. It is log of K2 over K1 equal to EA by 2.303 R 1 by T1 minus 1 by T2. This is the formula when 2 temperatures are given and 2 Ks are given. So now they are saying that rate of reaction quadruples means K also quadruples. So K2 is 4x when K1 is 1x. Log 4 equal to EA by 2.303 R. What should I put R? It's given in kilojoules. So I'll put the R in the joules, which is 25 by 3. I'll take 25 by 3000 now. Directly I'll get the answer in kilojoules. Okay. 1 by T1 minus 1 by T2, I can write T2 minus T1, which is 330 minus 300 by T1 T2, 330 into 300. Okay, solving for it, EA will be log 4, which is 0.60 multiplied by this whole thing comes up 2.3, 25, 330, 300. These all things come up. These all things go down. 3000, 330 minus 300, which is 30. Okay, simple. Now you can solve for it. Get the approximate answer because options are very far away. I'm directly getting it in kilojoule now because I put 3000. It will become kilojoule automatically. 30, 10 times. 10, 10, 20, 20. Then on the table of 5, I can cut it at 5 times. This will go 6 times on the table of 6.1. Okay. Then 5 times 2.3, which will be somewhere around 14 or so. 14.1, 1.4 into 33. So almost 1.2 into 33, somewhere around that. So around 38 kilojoule you will get. So I'm getting somewhere around 40. Just to solve, 38 kilojoule around we are going to get. Okay. So option 2 coming from here. Option 2 is coming from here. And going for the last question. The product A and B obtained in the following are: when you react alcohol with PCl3, 3OH go with phosphorus. So we'll get POH whole thrice. When it will tautomerize, it will give you H3PO3 as a different structure, both same formula, H3PO3. Whereas when PCl5 react, 1 mole only will react with 1 alcohol, 1 Cl goes with 1 H plus, rest 1 Cl goes with R. So in the PCl5, 1 Cl goes with R group, 1 Cl goes with H plus, rest 3 Cl remains with phosphorus and become POCl3. I hope all of you remember this final product as well. So B is POCl3, first one option is correct. Okay. All right. How was your paper, everyone? Just I'll wait for 2 minutes for your reply. How much marks you are getting in the chemistry, please tell me. Ishwar Babu, S3 key, you will get it on the Telegram channel. Just join the Telegram channel, you will get it there. How much marks you got in the chemistry today? All of you, those who are watching the video. Okay. I'll just wait for 2 minutes in case you guys reply. Otherwise, I will leave after few minutes. '88 Rehna. Okay. Lucky 125. Samvita 115. Lily 161. What's your name? Is your name Lily only? Good Lily. Good Maha. Kashi Tejaswini. Good 146 is also fine. Sir, Mother Mary 180. Jittu creation. So Mother Mary is getting 180 or you are getting 180? Others? Chalo, that's good. So any marks above 155, 160, I would say is a very good marks. Okay. So that's all for now. And I hope that you have understood. All right. Bye-bye. Take care. And now physics discussion will be started by Abhishek sir now. Bye-bye.
Hello everyone, good evening. So without wasting any time, let's discuss paper. Good evening. When physics will start? It has started. See. First question is, this is your magnetic field question. Tightly wound 100 tons coil of radius r, you can take 10 centimeter, carries a current of 7 ampere. Magnitude of magnetic field is required at the center. So magnetic field at the center we will write mu naught I by 2r, and as many turns are there, for every turn we will take this magnetic field for every turn. So total magnetic field will be n times mu naught I by 2r. Mu naught value is given here 4 pi into 10 raised to power minus 7. We are putting it. Current is also given 7. Since current is given 7, we will put pi as 22 by 7 to solve it. 2 into r is 10 centimeter, 10 minus 2 meter, that will be there, and number of turns is given here 100 turns, that will be multiplied 100 turns, right? So this is to 2, and pi we can put 22 by 7, so that will be 2 into 22 by 7 into 7, and all 10 power we will see here 10 raised to power minus 7, 100 can be written as plus 2, and then this is 10 minus 2 is minus 1, it will go up plus 1. 7 cancelled, it is coming 44 into 10 raised to power minus 4, but question options are in milli, so we will go with minus 4 is there, so it will be 4.4, 10 raised to power minus 3, that minus 3 will become milli, so that will be answer for this question. Just a single formula question it was, right? Second one is column matching, basic theoretical knowledge from magnetism and matter. Diamagnetic, ferromagnetic, paramagnetic, non-magnetic. In these kind of questions, first of all in NEET you don't have to see all the options, keep matching one by one. Like diamagnetic is there, we know that diamagnetic has negative susceptibility, and negative susceptibility means greater than minus less than 0, that is given here only. So A goes to 2. Now A goes to 2 is here and A goes to 2 is here, right? Then ferromagnetic, strongly magnetic, susceptibility is far greater than 1. So B goes to 3, B goes to 3 is here. So that is the right answer, fourth option, right? We can go by this also next, like paramagnetic it is here. C, more than susceptibility is positive, that has to be positive, little positive but not highly, not far greater than 1. That way we can match, but only matching the first two you can get the proper answer of this question. Next one is this. Unpolarized light beam strikes a glass surface at Brewster angle. Brewster angle is like this. When incident ray comes at a certain angle of incidence, polarizing angle or Brewster angle, so that two things happen here. First, reflected light is completely polarized and refracted light are partially polarized, and they are mutually perpendicular to each other. Perpendicular they are not asking in this question, only regarding polarization they are asking. So we will first look for reflected light should be completely polarized. So first option is saying both reflected and refracted completely polarized. We will not go with this. Reflected light will be completely polarized, but the refracted light will be partially polarized. Just knowledge based question is this, no numericals involved. So second option we will go in this. Next one is, in an ideal transformer, no power loss, right? Ideal transformer, what we write? E secondary upon E primary is equal to N secondary upon N primary, right? This is also called transformation ratio. But in question, if we will see, NP by NS is given. So we will write here reciprocal of this 2. This is secondary, this is primary. And they are asking ratio of what? Vs by Vp. That same E or V, whatever you write. So Vs by Vp. And here that will be 2. So answer will be, option will be 2:1. Again, this is also simple formula question, 2:1. Next one, logic gate. This question was little tricky, not tricky like it was there to give you two options like this that will force you to think that way and writing everything. But question is a very simple Boolean expression question. If you will consider this much only, you will get your answer. Moreover, you can go with like B is to not answer. If I'm simply looking at B, because less number of variables are there, so I will first check B. B is 0 and Y is 1. So obviously Y is not B. B bar I am checking 0, 1, 1, 0, 0, 1, 1, 0. Certainly it is B bar. Moreover, you can check these two, but it was not necessary because B bar is there, so that will be answer. Even if you will check this, it will not come. So B bar in these kind of question, generally we should start, if checking is the only way we are doing the question, we should start with the lesser number of variable option and then we will move on. So B bar we will get our answer. So no need to check these two. In a vernier caliper, n plus 1 division of vernier scale. So n plus 1 vernier is equal to, is coinciding with n division of main. And from here, 1 vernier is equal to n upon n plus 1 into main. And they are asking us simply what vernier constant is given. MSD represents 0, the vernier constant means they are asking you this. See, 1 least count, 1 MSD, vernier constant least count is required, minus 1 VSD. If you will put that value here, you will simply get 1 MSD minus 1 VSD is this. So it is 1 minus n upon n plus 1, and main scale division is given here in millimeter, 0.1 millimeter, but requirement is in centimeters. That unit conversion we have to do. So we will write that 0.1 into 10 raised to power 3, that is given in meter. But if, as I said, it is given in centimeter, so you will convert it and it will be here n plus 1 minus n, it will be 1, right, divided by n plus 1, and if we will convert that into centimeter, that will come here 10 raised to power minus 2, so that will be 1 by 100 n plus 1. That minus 2 is coming, minus 3 millimeter, centimeter you will convert, minus 2 will come overall, like minus 4 is coming here, that will come minus 2, and minus 2 means denominator will be 100. So n plus 1 in denominator in 100. Generally we can do this entire derivation I have told you. Generally we take the difference of these two, n plus 1 is there and n is there. We will simply take the difference divided by higher. So take the difference n plus 1 minus n and divided by the higher value. Who is higher value? N plus 1. So this is giving you 1 upon n plus 1, and that will be multiplied with main scale division, and from here we can directly get the answer, difference upon higher into MST. Okay, so this option will be fourth option. Next question. This is generally in the 5 year of NEET. If you will see this Young's modulus, this formula question generally get repeated every year. Maximum elongation of a steel wire. What formula here? Y is equal to F by A delta L by L, and they are asking us delta L. So what will be the value of delta L from here? Delta L is equal to FL upon AY. In this question, if you will read the data given, maybe you will look for force and you will look for area, but area is not given in the question directly. They have given the stress value, right? Means F by A value has been given. Where is F by A value? Dimensionally also you can check. Newton by meter square, F by A. So that value is given here also, F by A. Out of these two, one will be Young's modulus, one will be F by A stress. So we will read where is Young's modulus, where is Young's modulus is the second one, and elastic limit, that is stress value, is the first one. So in place of F by A, stress, elastic limit is a stress here, that is 8 into 10 raised to power 8. That we will write, okay, divided by Y. Y is given 2 into 10 raised to power 11. And length is given 1, that is numerically not going to make any difference. That we have to calculate 4 into 10 raised to power minus 3, and 4 into 10 raised to power minus 3 means 4 millimeter option we will take here. Okay. Okay. 120, 158. Okay. They're still in marks. This question, see, interference through white light, interference by white light, that is the question 1. In this we know that suppose in YDAC we are using white light, means various wavelengths we are using there. Central maxima is the point where all the wavelengths will interfere constructively, right? Because path difference, central maximum means path difference is 0. So all wavelengths are coming here and their interference is constructive because delta x is 0. So due to that constructive interference, at this point all wavelengths are superposing and giving you bright. So bright and all wavelengths superposing means central maxima will be white. Central maxima color will be white, right? But other fringes, if I will take, suppose first maxima I am taking or second maxima I am taking, at this all wavelengths are not constructively interfering, right? Because their path difference will not be following that. Like for constructive interference, what you say, delta x equal to n lambda. So this is not there for every wavelength. If I am not taking central maxima, I am taking first maxima, second maxima, maybe out of many wavelength 2, 3, 5 are interfering there. So they will have colored fringes. Maxima will be there, but colored fringes will be there, because for making white all wavelength must interfere, right? So that is only taking place at central maxima. So this is the characteristic of white light, कि central maxima will be white, other maxima will be colored. So that we will have to see in option. First one is saying there will be a central dark fringe. That is absolutely absurd. All bright fringes will be of equal width. Interference of equal width. Width also you are calculating, that also depending on lambda. Interference pattern will disappear, that is wrong. There will be a central bright white fringe surrounded by few colored fringes. That is the absolute answer of this question. White, and we use the white light interference for detection of central maxima. Like we are using monochromatic, so all bright will be equally bright, and you will not be able to detect where is central maxima. So this is why white light interference we do to detect the position of central maxima. Central maxima need not be geometric center. At any point where white fringe is being formed, that will be central maxima in this experiment. Okay. No question is there? Question, laws of motion question, very simple question is this. In these kind of question, we will first see friction is available or not. We can see there is no friction. So complete acceleration will be C. Acceleration, force upon total mass, and total mass is 6, and force is also given here 10. So it will be 10 divided by 6. This is your complete acceleration of system. Means A and B both are accelerating as a combined block with that much acceleration. Now to calculate this force between A and B, we can now choose system either A or B. So I am choosing system, suppose A, and A mass is 3 kg. What force we are applying on it? 10. What force B will apply on it? Normal, let's say N. But it is accelerating towards right because net force was towards right. On this, if Newton's second law we are using, 10 minus N equal to MA, 3 into A, right? A already we have calculated. That will be 3 into 10 by 6. That is equal to 2, and this is 5. Okay. I think this block is wrong written here. Question is 2 and 3. And in diagram it is written 3 and 3. So we will take A as 2 kg. Diagrammatically it is wrong printed. Question is right, 2 kg, 3 kg. So 2 kg and 3 kg, then this will be 5. And 10 by 5, then that will be 2 meter per second square. That is the value of acceleration. And if acceleration is 2, from here only, this is now not needed. 10 minus n equal to 3 into a value is 2, so that will be how much? 3 into 2, that is 6. Okay, and now you have to calculate normal reaction. Normal reaction will be 10 minus 6, that is equal to 4 Newton. That will be answer. Okay, that was a wrong mistaken point here. But how to do this question? Step 1, we will calculate total acceleration. Step 2, we will pick any of the block, either A and B, and their force diagram Newton's second law we will apply, and we will get the answer. Next one, this we have done already. Now see this question. In this question, simple one formula question is this, right? One formula question. What is that? Lambda is equal to h upon under root 2me. We all know this, right? It is mv, momentum, and like de Broglie, if you will write lambda equal to h by p, but we know that p squared by 2m is equal to e, and from here we can calculate momentum as 2m. But see, identify the quantities given in the graph. Basically focus on y-axis and x-axis. What is the y-axis? Y-axis is this, 1 by lambda square. And what is x-axis? X-axis is e. X-axis is e, right? Okay. Now see, how that quantity come? Lambda square is not here, so we will write lambda square is equal to h by 2me. And then 1 by lambda square, flip this, is equal to 2me by h. Now focus on y-axis and x-axis. What was y-axis? 1 by lambda square. This is our y-axis and this is our x-axis. So graph comes y proportional to x. And y proportional to x will be straight line passing through origin. In this question, many students have taken rectangular hyperbola, assuming lambda square and e are inversely proportional. But question is not asking us graph between lambda square and e. The graph is between 1 by lambda square, so we will take that way. Okay. Okay. See the next one. This is very simple again. Wheatstone bridge. Data also in this question is very easy. 5. Whenever we see 5 arrangement, one thing comes to our mind, ki maybe it is Wheatstone if ratios goes that way. And the 5th capacitance or 5th resistance, they should be connected between those 2 points where terminals are not there, means perpendicular to terminals, cross with terminals. Terminals are these points. This is A, this is B. So my 5th capacitance must be connected between those points where A, B are not there. So A, B are not there here. So it means it is Wheatstone bridge. 5th capacitance will go. If that will go, these two will be in series. 2 series, that means 1 microfarad. 2 series, this is also 1 microfarad, and 1 and 1 in parallel, we will use the formula C equivalent, C1 plus C2, 1 plus 1, and that will be 2 microfarad. One basic formula answer. Third one. Okay. See the next one. Now, this is your Lenz law question. I think 2 questions were there from Lenz law. See what happens here. This is a magnet, right? This is a magnet. And this magnet, south is going here. South end is going here. If south end is going here, it means according to Lenz law, this should oppose this movement. What is Lenz law? The direction of induced current will be in such a manner, it will oppose the cause who has induced it, right? So what is the cause? Cause is south pole is going towards the coil. So now south is going towards, so south has to be repelled. Who will repel south? South repels south, like poles repel. So we will have an induced south pole here. Induced south pole. Now see how the south pole will come. Whenever you have to induce somewhere south, it means it should be in what? Clock. And when you are inducing north, then it will be anti-clock. You see from here, here should be south pole, means you should see here in clock and that should be in anti-clock, right? So from here north is going away, so here also south will induce. Here also south will induce, and here also south will induce. Both side, here also south, here also south. This also should look clock, this should also look clock. That clock way, if we will think, so that direction will be from here, it will be AB, and this will be opposite DC. So we will look for AB and DC, because C is here, C end is here, and D end is here. So you have to see this coil is going like this. So current will go from D end and it will go like this. That will induce, that will repel south pole from here. So D to C, and this type it will be A to B. You can do this question not by like putting this. Suppose you are looking the coil from front, and from front you can look like this. This is the first coil AB, and then see where is the direction. This is the second coil CD, and then see the direction here. Both will be south here. So in CD, see the direction, and AB, see the direction. It will be AB and DC. So that should be your answer for this question. Okay. Next one, consider the following statement A and B and identify. First, read A. For a solar cell we know that this is the characteristic curve, and that is in fourth quadrant, right? So let's say what this is saying, IV characteristic lies in the fourth quadrant of the given graph. That is absolutely right. Okay. So A is correct, A is correct. B pe, reverse biasing PN junction diode, current measured in microampere, it's right, current measured in microampere, reverse biasing. But answer is right or not, that we have to read further. Graph of PN junction like this. So this is in microampere, we measure. If you will read question till here only, not read further, that will be a silly mistake. Because again question is saying due to majority charge carriers. But we all know that reverse biasing there is due to minority charge carrier, not due to majority, right? So if you are reading till here, you might feel both are correct, but that is an added statement here that makes statement 2 wrong, statement A right. So we will say A is correct and B is incorrect. Only incorrect in B is the majority charge carrier. It's not a majority, it will be minority. Okay, now this question, this is a little calculation, not that much difficult, but yes, it is a little calculative one. Okay, see what is the calculation here. First of all, focus on AC. Focus on AC beta. I am drawing again for you. That is A and that is C. If you will see on AC, it is grazing emergence. Grazing emergence means if you will draw normal, what angle is there? 90. If here is 90, it means R2, the second surface refracted incident angle R2, that is equal to, numerically equal to critical angle. Because one angle is 90, so R2 will be equal to critical angle. We also know that prism angle, this prism angle is R1 plus R2 equal to A. So if I will write R1 plus R2 is equal to A and R2 is already equal to C. So what it makes R1 equal to? A minus R2. R1 value we got, A minus R2. But again see in the diagram what is A? Prism angle is how much? Here it is given 90. So I can write this as 90 minus C, right? Now 30 degrees is the incidence angle. Use Snell's law, sin I is equal to mu sin R1. Here angle is R1 and that R1 is equal to 90 minus C. So that will be equal to mu cos C. We know that sin 90 minus C is cos C, and sin I is 30 degree and sin 30 degree is 1 by 2. Final step of the question, 1 by 2 is equal to mu cos C is equal to under root 1 minus sin square C, and sin C is 1 by mu. From here if you will solve, you will get mu equal to root 5 by 2. That is the way of solving. But if you will use ABT, like means Abhishek's a trick you will use. Suppose whenever refractive index question comes, mu question is coming, first I will see question is asking me refractive index of prism, right? It has to be greater than 1 because outside is air. So prism refractive index will be greater than 1. I will see my option first, how many options are greater than 1, and that also I will find only one option, fourth is greater than 1. I don't need to calculate it because all are less than 1, only 4 is greater than 1. That has to be refractive index of prism. That is a trick way we can solve. Method way if you want, that is the method. R2 is C, R1 is A minus C, and then Snell's law, solve, you will get mu. But as a smart solver, don't have to do that. If mu's two options will be greater than 1, I will go there. Otherwise, I will directly take root 5 by 2. Other are not numerically possible for prism. Okay. Okay. See next. This question. One of the easiest I must say in today's paper. But first mistake in this question, potential. Many of the students used the formula of electric field. Axial point, equatorial point, habitual of doing for electric field, and NEET beautifully has asked you potential. That is a simple mistake. If that potential hit your brain right at the right time, you will use formula this. Potential for dipole is Kp cos theta by r square. This is the dipole, right? And axial point we are looking at. So axial point from center on the axis at 2 meter. So theta will be 0 degree. Theta will be 0 degree. What you will use? V equal to Kp cos 0 degree is 1 by r square is 4. K is 9 into 10 raised to power 9. P is how much? P is given 4 into 10 raised to power 4 into 10 raised to power minus 6. And r square is already 4. 4 cancel. That is 9 into 9 minus 6 is 10 raised to power 3. So V is equal to 9 into 10 raised to power 3. That is absolutely right. Now students have taken here 2p because axial point 2p is there, and that way they have got wrong answer, because in potential 2p is not there. That is in electric field. Potential, it is given 2 Kp by r square. That is wrong. Potential is not 2 Kp by r square. Potential is Kp cos theta by r square. Axial point cos theta will be 0 degree. So Kp by r square. 2p is not coming in potential, and r square is not coming in electric field. So 2 Kp by r cube will be the formula. So that 2 is little misleading, but answer we will talk. So assertion is right, reason is false. Assertion is true, reason is false. That will be answer for this question. Okay. Got it? Now see the next. Moment of inertia. You know, in this question where the mistake has happened when I was interacting with kids here. You know the formula ml square by 12 is equal to moment of inertia. Right. Now question is asking you length. You are calculating L square. 12 into I. What is the value of I? I is given here. See, axis passing through midpoint perpendicular I is 24. So it is 2400. All are in centimeter. Then I will divide by M. M is given 400. Right? And here it will be 6. And 12 into 6, 72. But 72 is also in answer. So if you will calculate L square and take 72, that mistake can happen. L square is 72. If I will calculate L, that is root 72, we will get answer third. Third will be the right answer. Okay. Next one. Circuit question. If any kind of circuit is given, any kind of circuit, right? You first have to think current in the circuit. The rest everything will depend on current. Current is that mandatory in the circuit. So I will first calculate current for this, and current will be 10 divided by total resistance E by R. Total resistance will be 4 and 1, 5. That will be equal to 2 ampere. And then since one battery is, one cell is there, its potential difference if we are calculating, that will be E minus IR, and that will be 10 minus I is 2, R is 1, and that will be 8 volt. That is the first option. Simple question, one formula only. Okay. See the next one. This question also, data is there, but calculation is not required in this particular question also, right? No calculation required. We all know the formula at 1 by lambda rz square, or proportional to 1 by n final square minus 1 by n initial square. You can calculate all. But you also know that as much as that difference, more is the difference. So more is the energy, and more is the energy, less is the wavelength. Less is the difference. Less is the difference means 3 to 2. Less is the difference. Here difference is of 1. Difference is of 2. Here difference is, there is no difference. Difference is of 4. So less is the difference, more is the energy, right? That way I will go that, I don't know this data 2 and 2 is there or not, or something else in the data, maybe typing mistake is there. So 3 to 2, that will be the least of difference, and least of difference will be the maximum of wavelength. So I will go from here. You can calculate that way also, or you can logically, empirically also solve it. So more is the difference in orbital shells, more energy you have to give to electron to go there, or transition, emission, absorption energy will be that way more. But energy is more, so wavelength will be less. Energy is less, wavelength will be more. So I will go A to third, highest wavelength. And A to third, when I am looking at, I don't have to think other also. So minimum, maximum you can decide in this question. Minimum only if you are thinking A also, the highest one, you are only getting one option, fourth will be answer. If you want to calculate it full, this is the formula for you for calculating, but it is not at all required. In most of the time in NEET calculative questions, direct calculation you should not start. See some other tricks are there or not, other logics are there or not, because that will be there in most of the time. So we can solve this question with that logic, that less is the difference, less is the energy, more is the wavelength. Okay. Go on next. Now photon theory question, right? What are the characteristics of photon? If c is the velocity of light in free space, the correct statements about photon discrete energy level, we all know this. Energy of photon is h nu. One photon energy will be h nu, bundles it will be nh nu quantization. Velocity of photon is c, that is also right. Momentum of photon is u by c, and u here is h nu, so h nu by c, that is also right. So A, B, C are right till now. In a photon-electron collision, when photon-electron collision that you study in photoelectric effect, Einstein photoelectric effect. So photon is colliding with electron and it is transferring its momentum and it is transferring its energy. Total energy and total momentum are conserved. That is also okay. Photon possesses positive charge. We all know that photon has no charge. So that is absolutely directly wrong statement there. So photon theory deals with above 4 statements. So we will go with A, B, C and D. So A, B, C and D is the fourth option. That will be the correct option for this. Okay. Hello Aishwarya. Okay, see the next one. This one is again a direct question. Just you have to know how much transition will go place. 290 is there. Alpha is helium nucleus. What is alpha? Helium nucleus. This is 4. This is 2. So when you are taking one alpha out of it, what will happen? 4 mass number will be reduced. So 290, 82, that is going to be 286. And here 2 you are taking from here also. 82 to 2 you have taken, that will be 80. And you have reached at Y. Okay. Then E plus is going. So plus will go, it will affect the atomic number Z. And it will not at all affect because no mass number is going. Mass number means nucleons. And electron is not related to nucleon, that is neutron, proton, right? So I am not taking here nucleons, E plus I am taking, so it will be 286, but since plus is going here, so 1 plus will be reduced and it will be plus is going. Then beta is going again, it will be 286 and 80. And again one more electron is going, so it will be 286, minus is going, so plus will be increased to 81. So 286 and 81 we will look for. Only mass number is changing in alpha whenever nucleons are exchanged. Other electron, beta plus, beta minus, in all this, beta minus is going there. It will only take your, affect your Z, not affect your A. So A is not getting affected, A will remain after this 286 and accordingly Z will affect. That will be final answer for you. Okay. Now this again mechanics question came. At any instant of time, displacement is given, right displacement, that is 2t minus 1. That is your displacement. And force is given constant, force is not time dependent, and that is 5 Newton. So what is the value of instantaneous power at a particular time what is the power, but in this question if I am calculating velocity, that is ds by dt, I am getting it 2. It means uniform velocity is there, so at every instant power will be same only. Power is not depending on time, so power will be here F into V. F is given, V is 2, that will be 10 units. So 10 units is third option. We will go with the third option. Hello Madhu. Okay. Now this question is based on your Boolean expression. Why we have to calculate in what gate? Logic gate. In AND here this is NAND and this is NOR, but these are calculated, these are joined, inputs are joined, so that becomes your completely NOT gate, and this is also becoming your NOT gate. So basically this input A NOT is A bar and B NOT is B bar, and here they are in NOR gate, it means OR, and here is NOT, that is this. So OR may, what is this? A plus B, A bar plus B bar, and then NOT. De Morgan's theorem we will use in this, and De Morgan theorem is telling us A double bar into B double bar. A double bar is A, B double bar is B, and A into B is becoming our AND gate. Okay, A bar, A naught, B naught, A bar, B bar, they are adding, and whole bar, NOR gate. So A double bar dot B double bar means A dot B, and that is the AND gate. Okay. Okay. Next question. Mass of planet is given. It is 1 by 10 that of the Earth. So mass of the planet is mass of Earth divided by 10. And its diameter radius will change by the same fraction. So radius of planet is radius of Earth divided by 2. The acceleration due to gravity on that planet, small g equal to capital G, mass of planet, the radius of planet square, that is g. What is mp? mp is me by 10, and what is rp? re square by 4 square here. So gme by re square, that is 9.8 into 4 by 10, and that will be 3.92 meter per second square. Okay. Next question. See this. Atoms are electrically neutral. Atoms are electrically neutral as they contain equal number of positive and negative. That we know from 6th and 7th class, right? Atoms are electrically neutral, right? So this is absolutely right. There is no diciness in this. Statement 2 has little diciness. Atoms of each element, if you will see, there are two things here. NCERT also has changed that statement. Earlier, the old NCERT had the each element atom of each element is stable. Our new NCERT is saying little different, most of the. But as far as the instability is concerned, instability is mostly in nucleus due to radioactivity. But nucleus is inside atom, so if nucleus is unstable, atom is also then unstable. So that way we can take it, but as of now, I will take it both statements are true that we have given in the keys also. Both statements are true, but there is a diciness in this one word, each and most of the. So most of the atoms are stable, few are not stable, and due to obviously because of radioactivity, most of them are not stable. But in radioactivity, nucleus is the major source of that, not atom. So in this question, we will take that both statements are correct, right? That we will take. From here, next Abhilash sir will discuss all the questions. So hope you have done well all paper today. It was little tricky, but not on difficult side. But yes, if you are doing all 180 questions together, physics was little tricky. Okay, all the best to all of you. Good luck.
Hey everyone, I think I'm clearly audible and visible to all of you. Yeah, first of all, many congratulations here. Today, most of you have won the battle, and the people who are still connected to see the last, the last time video discussion to get to know where the mistakes are there. I really appreciate you all. The reason is what I always say, that keep learning from your mistakes, and learning has no end here. Okay, so even if this was so called the last battle you had after this long journey of NEET preparation, this was actually one of the first battles you probably have won as far as your entire life is concerned. So definitely there are a lot more to come, and you need to enjoy the learning. So the journey is somewhat the most beautiful part of every process, rather than the major destination you are planning to reach. So I hope you have enjoyed the learning with us, and you yourself have prepared very well, and we'll keep learning together here. So I won't take much time because it's already very late night, and you probably had a very hectic day, and you have exhausted yourself too much, put your entire energy into the exam. So yeah, but before I start the discussion, I would add on one more thing for all the people who have not got who are not getting as per the answer key the expected marks, or you're thinking that you might not get the expected ranks and all. I would definitely say, we as humans plan very well. Smart people plan very well what they are going to do in the next 4 or 5 years of life. But ultimately, trust me, it's a human plan. So what God plans for all of us is far much better in comparison to that. So if you have given your best, the efforts were under your control. If you really have given your best, then be satisfied. Very soon, life is going to give you bring a lot of magic. Make sure you embrace them. Make sure you welcome those magical things with a big smile. Okay, so let us start with the discussion. Yeah, hello everyone. Chaliye. So this is a problem which came from rolling motion officially, and apparently we used to say that rolling motion has been deleted from the NEET syllabus, and this was not a too much technical question based on rolling motion. It was a very simple problem here. The problem was that they are giving you that a wheel is in pure rolling motion, it's moving in the forward direction, and you have to tell that which of the points will be having relatively greater velocity. So naturally, if rolling is nothing but, it was based on a very simple thing, that rolling is nothing but a combination of translation. It can be written as a translation effect plus a rotational effect. Okay. So as a result, so if you see the translational effect, you will find that each point on the wheel will be having a V0, or velocity of center of mass velocity. Each point will have V0, V0, V0 and V0. That's the property of translational motion, that each and every point is going to move in a parallel path, or you can say they will be having the same velocity. And as far as rotational motion is concerned here, pure rotational motion, every point will have the same omega. So this will have some r omega, linear velocity component in the forward direction. This will have r omega in the backward direction because of the clockwise rotation. This will have r omega in the downward direction, and this will be having r omega in the upward direction. So if you basically compare the velocity of P as well as the velocity of Q, the P point will be having V0 as well as r omega in the forward direction supportive behavior. And if you see the bottommost point, that's the point of contact, which is also called as instantaneous center of rotation or point of rotation. It will be having the V0 velocity in the forward direction, but r omega will be having a backward. Okay. So pure rolling mein naturally V0 and r omega are equal, but even if we didn't learned a lot of rolling motion because of the confusion in the NEET syllabus, then also at least you can answer this problem on the feeling based, right? How? The topmost point will have V0 also, r omega also, supportive in nature. Bottommost point or point of contact will have V0 forward but r omega backward. So naturally they are in opposing sense. Hence, correspondingly you can say that P point will be having a more velocity, because V naught plus R omega supportive in comparison to the Q point, which will have opposing behavior. So I don't want to get into too much mathematics, plus, minus, angles, this, that, which one is at rest and all, but feeling based, you can simply answer that. Okay, so the correct option is P point has, point P moves faster than point Q. Okay, the upper point will be moving faster in comparison to the lower one. All right, let's move on to the next one. A particle moving with, and you can put your doubts in the chat. Okay, and yeah, congratulations, you got good marks in chemistry too. Chemistry was a bit easy, you know, this year, and physics was, I would say, relatively more standard paper we got for physics. Naturally, in comparison to the last year, it's just my personal opinion, it was a bit on the higher side because the questions were relatively good. Okay, so yeah, coming back to the next one, it's quite a confusing problem. Particle moving with uniform speed in a circular path. I would technically say uniform speed in circular path means what we call commonly as UCM. The nickname UCM is given for, that is uniform circular motion. So whenever the uniform circular motion comes, people feel that acceleration is equal to V square by R and V is going to be constant. So naturally acceleration is going to be constant. But remember dear, acceleration and velocity both are vector quantities. Okay, so as far as the constant and variable behavior is concerned, you don't have to just stick with the magnitude, you need to think about the direction too. So if I talk about the direction, a particle is moving in a circular path. Again, you could have answered feeling based. You need not to know a lot of circular motion for this. Suppose it is at point M right now. So this is the direction of velocity, Vm cap, and this is the direction of acceleration, Am cap. Only centripetal acceleration is there. And suppose the particle reaches at point N somewhere. So this is the tangential velocity Vn cap, and this is again directed towards the center, An cap. So naturally you can see that with the passage of time, the position will be changing, the tangential direction of velocity will keep on changing, as well as the radial direction of acceleration too keeps on changing. All right. So the correct option is it will be having a varying velocity as well as varying acceleration. All right. Let's move on to the next one. A thin flat circular disc, very, very simple and very nice question based on surface tension. Yes, that's a part of fluids. Okay, fluid mechanics, generally students are, a bit reluctant to do the topic, but entire year, if you remember, we used to say that, fluids curlo, do the fluids, because most of your competitors will eventually decide to leave the fluids, but the questions from the fluids are going to be very easy. Okay. So a thin flat circular disc of given radius, we will definitely convert it into SI units during the calculation, is placed gently over the surface of water. If surface tension is given, then the excess force required to take it away. Why they are saying excess force? Because naturally whenever you want to uplift something, definitely you have to overcome its weight. Okay. So what additional they are trying to ask other than weight, we will see. If suppose that is placed initially on the surface of liquid and you are trying to uplift it, so definitely there will be a force due to surface tension which will try to bring it back. You need to overcome that. So here force due to surface tension will be given by surface tension into free length. Free length is that length of the body which is in contact with the fluid. So since it's a disc, so what free length, peripheral length will try to be in contact with the liquid? That's indeed 2 pi r. So T into 2 pi r. Fine. Let's keep the values in SI units. T can be written as 0.07. Let me write it as 7 by 100. That's 2. Pi can be written as 22 by 7 in a hope that definitely 7 and 7 I can cancel and we can land on to relatively simpler conclusion and calculation. Pi r. R is 45 by 10. And do remember it's in centimeters, right? So we would like to divide again by 100 so that we can convert it into meters. Okay. Now 7 will get cancelled. I think 22 2 multiplied with 45 you will get 90. So it is 22 into 90. The options are in millinewton and all. So I think I'll try to keep it all together, because if it is in denominator like 1000, you can write it as milli simply. So it is 2 into 45, that's 90, 22 as it is, by 100, and it is millinewton. So see, you can simply cancel it out. And 22 into 9, 18, I think 198 by 10 millinewton, or you can write it as 19.8 millinewton. Let me check any option. 19.8. I guess I'm getting option 3. Okay. So it was not based on any complicated or approximation required based calculation. Simple problem, simple calculation. Okay. Chalo. Yeah, Abhishek, Rama, yeah, I can see all of your comments. Let me, complete the video discussion, and definitely then we can talk about the marks. And yeah, we posted already all the answer keys were already posted in the groups and all, so you can verify from there, right? Then we will definitely get in touch regarding all the queries. Let me complete the discussion first, right? So the next question is, in a uniform magnetic field given, again that's already in Tesla means SI unit, that could have been in Gauss. We could be requiring to convert it into SI, but anyways that's not the requirement in the problem, so it's a simple one, already in SI unit. Then they are giving you 20 oscillations in 5 seconds. 20 oscillations in 5 seconds means I will say 20 oscillations. That takes 5 seconds. This implies one oscillation will be 5 by 20 seconds, or you can say 1 by 4 seconds. Naturally, you can write it as the time period. Time period, capital T. Now they are expressing the value of magnetic moment in terms of x into something, where you are required to guess the value of x. Alright. So we will say here, what do we know? We know time period value as 2 pi under root I by mb external. Is it fine? Now, time period is given to you as 1 by 4. That is equal to 2 pi as it is, because if you see all the 4 options are in pi square. So let me keep the pi as it is. Under root of moment of inertia is given to you. Yeah, that is given to you already in SI units. See, kg meter square. So that's 9.8 into 10 to the power minus 6 by magnetic moment. You are supposed to calculate B external. B external is given as 0.049. Let me write it as 4.9. 4.9, 2 decimals I am shifting on the right side into 10 to the power minus 2. Why you are preferring to shift 2 decimal places on the right side, sir? Because I can see that 9.8 is a multiple of 4.9. So it will be cancelled by 4.9 at 2 times. So let us do one thing together. Let us send this 2 pi in the denominator so that we will get 1 by 8 pi, because it's ultimately the calculation now. And I would like to do the square in the same step. So what I'll be having here is 1 by 4 into 2 pi means 1 by 8 pi, and let us do the square. So 1 by 8 pi into 1 by 8 pi, and that will be equal to 9.8 will be cancelled by 4.9, and that's what you will get 2. But square has been done both sides, so root will go off. 10 to the power minus 6, and that's 10 to the power minus 2. I guess that will be 10 to the power 4, minus 4, and that's m here. So if I try to write m, I will get 8 into 8, 64 into 2, 64 into 2, 128 into pi square into 10 to the power minus 4. Again, 8 into 8, 64 into 2, 128 pi square into 10 to the power minus 4. But they are giving it in 10 to the power minus 5. So if you forcefully try to convert it into 10 to the power minus 5, obviously you have to write it as, multiply by 10 and divide by 10. So 1280 pi square into 10 to the power minus 5. I suppose if you compare, this is the data you were searching for. So the correct option is 1280 pi square. Is it fine? You can just have a look for the calculation. And I don't want to say that, okay, you can do the calculation on your own. No, if you have noticed from the starting of this session, every teacher is in front of you and we are telling you the problem starting from the base up to the, starting from the scratch up to the completion of the problem. All right. So that you don't have any kind of confusion and don't get any sense of disappointment or any sense of achievement in between. Try to learn it if you have already joined the session. Okay. Two problems this way, that way can happen in the exam here. It's a national level exam. Moreover, your career deciding exam. So a bit of, being anxious and bit of examination pressure and then one or two silly mistakes, it's completely fine. Being students, please give yourself a bit of liberty to do some small mistakes today. Okay. And definitely, you are going to be the future leaders. All right. Now, taking you to another one. Okay, if you think that there are a few mistakes in the answer key given, okay, we can again have a look, because different answer keys would have been there by this time, and if one or two mistakes are there, we can again have a discussion. Don't worry. But here, whatever is being discussed for you, you can take this as a reference and then have a quick calculation. You'll find it correct. Okay. Next problem. Two bodies A and B of same mass undergo completely inelastic collision. Completely inelastic means perfectly inelastic collision. The bodies are going to stick together and move. And they are saying A and B of same mass. So one is M, another one is also M. The body A moves with velocity v1. Initially, body A moves with velocity v1. And what about body B? While body B is at rest. Okay, body B is not moving, it is at rest. Fine. The velocity of the system after the collision. So they are saying perfectly inelastic collision, E equals to 0. Bodies will stick together and move. In that case, if they have been stuck together, this is MM. Let us suppose they are moving with VC or V2. In fact, this is V2. So here, if you apply conservation of momentum, you'll have mv1 plus m into 0. That's gonna be 2m into v2. So see, m got cancelled out. You got v1 is equal to 2v2. Now let us see, examiner is asking us what? They are asking the ratio of v1 by v2. So if they are asking you the ratio v1 by v2, correct option is 2:1. 2:1. Is it fine? I hope it's clearly visible for you. All right. Chaliye. Let me take you to another one. If x equals to 5 sine pi t plus pi by 3. Now it was a very simple question. Every student knows this question. The probable mistake is that they are deliberately triggering you in some set of papers. In every set, the option sequence is not like that. But in some set of paper, they are deliberately triggering the people to, choose 5 centimeters. And in some set, it was 5 centimeter 2 seconds, which was a probable answer. But actual answer will be corresponding to meters naturally, because the equation is given in meters. Okay. So the position, amplitude, everything will be definitely answered in meters. Let's see. So they are saying, what is the amplitude? Definitely amplitude is going to be 5. What about the time period? Suppose the coefficient of t is omega. So t will be equal to 2 pi by omega, but omega is supposed to be pi, coefficient of t, pi got cancelled, answer is 2. Answer is 2. So amplitude is 5 meters and the answer is 2 seconds. Option 4. Hope that's okay. All right. Move on to the next one. The quantities which have the same dimensions as those of solid angle. Everyone knows solid angle is dimensionless. Okay. So if it is dimensionless, you have to choose that option which contains the remaining physical quantities also to be dimensionless. Which are those? Strain and angle. Strain and angle both are dimensionless. Okay. So the correct option is option 3. Like dimensional analysis, say since last 1 to 3 years, you are every time getting very easy questions. And if you notice, somehow they are based on angle, solid angle, steradian, radian, like that. Okay. Previously also you got the problem like dimension, it has units but no dimension, no units but dimension, like that. Okay. Let's move on to the next one. A thin spherical shell is charged. If you remember, thin spherical shell. Thin spherical shell, it has to be metallic and conducting in this, inside region. So it is charged by some source. So you see the entire charge is distributed on the surface. Inside region where there is no field, no charge. So V at the center will be equal to V at the surface will be equal to V at any inside point, isn't it? What they are asking actually. A thin spherical shell is charged by some source. The potential difference between 2 points, yeah. So V at any inside point, So everywhere the potential is going to be same. This implies potential difference, VHL, potential difference, higher, lower, that is going to be 0. Way 1 of understanding that. Way 2, easier understanding. Way 2, what is the electric field existing between C and P? Is there any electric field that existing between C and P? Existing between C and P. No electric field. So if there is no electric field, the potential difference is also going to be 0. Why, sir? Because if there is a potential difference, electric field will definitely exist from higher potential to lower potential. Alright? Chaliye. Doing the next. Next problem is, a bob is whirled in a horizontal plane by means of a string with an initial speed omega. So there is the bob. It is, suppose, m mass, L length string. So it is whirled in a horizontal circle with angular speed omega. So if you want to talk about the tension, what they did is, they doubled the omega. Initial angular velocity was omega, now it is 2 omega. Initial tension is T, final tension. They are asking, what is the value of final tension? So what you can say here, you will say tension will be equal to centrifugal force. So here, or you can say in other words, tension will provide the necessary centripetal force, m omega square r. Now if naturally you are going to make it double, 2 times. So clearly you can see that T is directly proportional to omega square. If you are making it 2 times, this implies T final will be equal to 4 times square, and 2 ka square 4 times T initial. Hence the final answer has to be 40. Option 4 is perfect. Alright. Chaliye, move on to the next one. A wire of length L, that is a very interesting question here. Very easy question. A normal 10th class student can also do that if you talk about the concept involved. But under the NEET pressure, and that too first series, then parallel, then series combination, like that type of question it is. I hope that you would have tried this question in the exam. So concept involved is not so big, but definitely step-by-step involvement is there. So I'll do it like in a kiddish way. A wire of length L and resistance 100 ohm in total is divided into 10 equal parts. So I will say resistance for each part is going to be 100 ohms by 10 parts, and hence resistance of each part will be equal to 10 ohms. Resistance of each part is equal to 10 ohm. Next, the first 5 parts are connected in series. Stop here. So you will say R series, 5 parts. So 5 parts of 10 ohms each. Answer is 50 ohms. While the next 5 parts are connected in parallel. So stop here. Now R parallel will be equal to 10 ohms by 5. By series mein resistance equivalent becomes n times multiplied. Parallel mein it gets n times reduced. So that's gonna be 2 ohms. Now the two combinations, these two combinations are further connected in series. So now these two combinations are further connected in series. So naturally you have to add them up. So final answer will be 50 ohm plus 2 ohm. Definitely the answer is 52 ohms. Hence option 4 is going to be the correct one. Alright. Done? Yeah. So decay question is out of the syllabus, and any add mark by NTA. I'll come to that, don't worry. I'll come to that. And I would say radioactive decay, alpha, beta and gamma decay. Definitely that was not in the syllabus. But once you study the stability of the nucleus, then in studying the stability of the nucleus, you have a graph, a relative number of neutrons and protons. So there we are supposed to understand the basic meaning of beta decay, beta positive decay and all. I mean, in all of the batches here in Sri Chaitanya Gosala campus, I'm taking almost all the batches of boys campus, girls campus, and there in all the batches, I'm damn sure it has been taught. I myself have taught it during the teaching as well as during the revision time. So it's a very important graph here, relative number of neutron and proton, and there we do discuss regarding the beta decay, beta positive decay and all. Okay. So what NTA will do for that, I will prefer to reserve my opinion on this. But as far as whether it is in the syllabus or not in the syllabus, we can't surely say that no, it has to be deleted like that. That's my opinion, because we have taught here. If you were taught, if you have, your teachers would have taught you like this, then you are supposed to do that here. Because if a student is aware of the syllabus, the teachers also obviously here. But we teach you for some reason, and see that's why we say follow the guidelines properly, follow the guidance also throughout the year from your heart, then definitely you will succeed here. Anyways, we'll talk about it and we will see what NTA does with that. I just shared my personal opinion. So yeah, let's talk about the next one. Again, it's a very simple ideal gas equation based question. So they are just talking about which pressure will be higher, which pressure will be lower, like that. So I can just directly compare with the slopes. I will say we know PV equals to nRT. The only difference is in Charles' law and all those, we get a VT curve generally, VT curve. But there what they are giving is TV curve. No issues. I will say T will be equal to P by NR into V. So if I compare it with y equal to mx, I can see slope m is equal to P by NR, which means directly proportional to pressure. Use this logic. Slope m is directly proportional to pressure. So higher is the slope, higher is the pressure. As you can see, the maximum slope is of P1. So definitely P1 is greater in comparison to P2, and that is greater in comparison to P3. So the correct option is P1 is greater than P2, greater than P3. Very simple logic. Okay. Let's move on to the next one. Nice question. Nice question from Basics of Electromagnetic Waves. The question is very simple. It says that there is a parallel plate capacitor. They want you to relate the conduction current and displacement current. So we know as per the continuity of current, displacement current plus conduction current is always equal. And in the capacitor plate arrangement, whatever is the conduction current outside, exactly same as the displacement current between the plates of the capacitor. Magnitude is also same at every instant, as well as direction is also same throughout. So you will say displacement current of same magnitude as that of conduction current will flow, and that too in the same direction. So displacement current of magnitude equal to I flows direction opposite to that of I. Wrong. Displacement current of magnitude greater than I. Wrong. There is no current. Wrong. There is no conduction, but there is displacement current. So displacement current of magnitude equal to I flows in the same direction as that of I, conduction current. All right. So fourth option is there. Next is the property which is not of an electromagnetic. Another question from EM waves. Two questions like every year. Which is not of an electromagnetic wave. Focus on this not. Okay. See which option is wrong. Okay. So they travel with a speed equal to this. True. But that's not your answer. They originate from charges moving with uniform speed. That's wrong. And hence that's your answer, because everyone knows accelerated charge is a source of EM waves. So uniform speed is obviously not an accelerated motion. They are transverse in nature. True. The energy density in electric field is equal to the energy density in magnetic field. That is also true, which means they want to say half epsilon naught E square will be equal to B square by 2 mu naught. Everyone knows that's a common property. Okay, so you had to choose the incorrect one. That's what we selected. Next one, choose the correct circuit which can achieve. Do remember, it is not a balanced Wheatstone bridge, but you have to choose that option where even there is a probability to achieve the Wheatstone bridge. So if you notice, you can as of now directly eliminate two options. For example, if you see second figure here in this particular set, options may be different in different sets. Be careful. This is actually reverse bias. This diode is reverse bias, and if you see this one, option 4, here also the diode is reverse bias. So basically both these circuits are similar, and obviously two options cannot be your answer. So technically these two options can be simply deleted. Now coming back, I'll explain you the logic also, how we get to know. I mean, this is just an elimination approach, but anyways, I'll give you the idea how to prefer the correct answer. They say which can achieve, which means P by Q must be equal to R by S. So here if you see 10 by 10 means it should be 1:1. So R by S also it should be 1:1. But irrespective of the forward bias resistance, don't consider it to be an ideal diode. Irrespective of the forward bias resistance offered by the diode, obviously the parallel combination will give you less than 5. P by Q is 10 by 10, 1. R by S you need 15 by 15, no. But the parallel combination will give you an answer smaller than the smallest one. So definitely this can never give you 15. This can never give you 15. But if you notice here, then if possible, if forward bias resistance offered by the diode is 10 ohm. Don't consider it to be an ideal diode. If forward bias resistance for a real diode offered is 10 ohms, then definitely 10 plus 5, 15 at least can have a probability to be equal to this one. Because the property of series combination is that the resistance equivalent will be larger than the largest one. But here it has to be smaller than the smallest one. So less than 5 can obviously never be equal to 15. But greater than 5 at least has the probability to be equal to 15. Okay, so they are just asking you can achieve. Okay, that was one of the interesting problems that came in today's paper. Alright, let's move on to the next one. If the plates of a parallel plate capacitor connected AB battery, connected to a battery maybe, this has to be connected to a battery, typing mistake, are moved close to each other, then what will happen? So, dear plates are connected to a battery are moved closer. So the value of capacitance, which is epsilon naught A by D. So if D is decreased, if the plates are moved closer, then definitely the D will decrease and the capacitance value will increase. But battery is still connected, so V final will be equal to V initial. Q final will be equal to C final V final, where this is same but C final has increased, so overall Q final will also increase. And U final will be equal to half C final V final ka square, C final V final square. V final remains same because battery is still connected, but capacitance increases, so overall energy also increases. Okay, so let's read the option. The charge stored in it increases. Yes, Q value is increasing. The energy stored in it decreases. No, it should increase. Its capacitance increases. Yes, it increases. The ratio of charge to the potential. Ratio of charge to the potential means capacitance, Q by V or C. They say they remain the same, but as we have seen, the capacitance increases, so this is also wrong. The product of charge and voltage Q into V is ultimately nothing but the part used in energy. That should increase, This is also true. So I think it's true, false, like that. So A, C, E, I will say A, C and E only are correct. A, C and E only are correct. Alright, explanation is already started with the explanation itself. Is that fine? Alright, chaliye, let's move on to the next one. A force defined by alpha t square plus beta t acts on a particle at a given time t. The factor which is dimensionless. So if you see, suppose I have to write, I will say dimension of F by homogeneity, that will be equal to dimension of alpha t square plus, that will be equal to according to homogeneity, dimension of beta t. Okay, so if you express it that way, or which they are asking dimensionless. So from here you can write dimension of alpha. Dimension of alpha will be f by, like dimension of f by t square. So that's gonna be mlt minus 2 by t square. So you will get mlt minus 4. And dimension of beta. So that will be mlt minus 2 force by t. Beta t is f, so beta will be f by t in terms of dimensions. So that will be mlt minus 3. Now you need a dimensionless thing, okay? So suppose we do alpha by beta. So mlt minus 3 will be cancelled and you will be still getting a t minus 1, which has to be reduced because you need a dimensionless. So in order to remove the t minus 1, what you need to do? Sir, let us multiply it with alpha. So let us multiply it with t. See, if you divide them directly, you will get a t to the power minus 1. If you want to remove the t to the power minus 1, what you are supposed to do? Just simply multiply it with t. So the correct option should be alpha by beta into t. So alpha by beta into t, I think fourth option, basically alpha t by beta. Alpha t by beta, fourth option should be that option, which will give you a dimensionless thing. Okay, that you can directly comment after seeing the dimensions of alpha and beta. Next is a metallic bar of Young's modulus. Young's modulus is given. Let us see what data is given. Young's modulus is given to you. Alpha value is given to you. Length value of 1 meter, that is given to you. Delta theta temperature change 0 degree Celsius to 100 degree Celsius given to you. Area of cross section 10 to the power minus 3 meters square given to you. The compressive force developed in it is, they are asking the value of F. They are asking the value of F. So what you can do here, you can write F will be equal to YA by L into delta L. Okay, YA by L into delta L. Or you can write it as YA delta L by L, which can be written as alpha delta theta. YA, this can be written as alpha delta theta. I think you can substitute all the values there and then you can easily get your answer. Okay, you want to check? We can. 0.5 into 10 to the power 11. It's already in SI unit Newton per meter square. Then the area is 10 to the power minus 3. 10 to the power minus 3 already in SI unit meter square. Alpha value is 10 to the power minus 5. That will be per degree Celsius or per Kelvin, the change corresponding to 1. That's going to be the same. And delta theta 0 degree Celsius to 100 degree Celsius. That's going to be 100. Okay, 0 to 100, the change is also 100. So if you see that carefully, what we will get? We will get 0.5 into 10 to the power. See, that's 11 plus 2, 13 minus 5 minus 3 minus 8. 13 minus 8, that's 5 newtons. 0.5 into 10 to the power 5 Newton, but they are giving 10 to the power 3, 10 to the power 3 like that. Since the order of magnitude is same in all the 4 options, you can multiply it with 100. Remaining 100 you can multiply and write the 10 to the power 3 Newton. So if you multiply the 0.5 with remaining 100, definitely you are going to get 50. So the correct option is 50 into 10 to the power 3 Newtons. Alright. Yeah, let's move on to the next one. A small telescope, easy question. A small telescope has an objective of focal length 140 centimeters and an eyepiece of focal length 5 centimeters. The magnifying power for telescope for viewing a distant object. Distant object means they're asking you the normal adjustment. Magnifying power in normal adjustment is FO by FE. Definitely it is having a negative sign which shows the inversion, but they are just asking you the magnitude. So FO is given as 140 centimeters. FE is given as 5 centimeters, hence the answer will be 28. A very simple formula based NCERT problem. Okay. Next, an iron bar of length L, that's an interesting one. I like this problem. An iron bar of length L has a magnetic moment M. It is bent at the middle of its length such that the two arms make an angle of 60 degrees. So suppose that's the north pole and south pole. It is having a pole strength of M and M here, small m here, and length is supposed L. Then here you will write the magnetic moment capital M as pole strength small m into L. Okay. Now what you are doing with it is you are bending it into two halves. So definitely both the halves will have their own poles and the pole strength will not change here. Pole strength will not change. Obviously the the part which contains the north pole will induce south and the part which contains the south pole will induce north, like that. Okay, you'll have this. Is that fine? They are asking you the new magnetic moment here. Okay, an iron bar of length L has a magnetic moment M. It is bent at the middle of its length such that the two arms make an angle 60 degree with each other. Okay, the magnetic moment of this new magnet is, so if you bend it at the middle, you will have north and south here, you will have south, north here. Both the magnetic moments will be, I would say, M by 2 and M by 2, and what angle they are making with each other is, interestingly, south to north, one magnetic moment you are having here, south to north, another magnetic moment you are having here, and which you can, extend it like this. So definitely you have one magnetic moment M here, another magnetic moment M here, and the angle between them, if this angle is 60 degrees, suppose this angle is going to be 120 degrees. So it is m by 2, rather m by 2 here, m by 2 here, and at 120 degree the resultant will also be m by 2 only. The resultant will also be m by 2. Either you think it this way, way 1 of doing it, see, when f0 and f0 acts at 120 degree, definitely the answer is again f0. 90 degree pe root 2 f0, 60 degree pe root 3 f0, 120 degrees pe again it is f0. Okay, so m by 2, why m by 2, sir? Because the length has become half. If the magnetic length will get half, definitely the magnetic moment, this length will get half, magnetic moment will also be half. Way 1 of doing that. Chalo, let us see way 2, a relatively easier approach also to think. So if you see this type of figure, now if it is this length is also L by 2, this length is also L by 2, and this angle is 60 degree, if you notice the separation, if you try joining them, then it's gonna be an equilateral triangle. L by 2, 60 degree. So this separation effectively will also be L by 2. So M effective nu will be equal to pole strength into L effective. So that will be M into L by 2. Again the answer is capital M by 2. So there have been both approaches, right? There would have been these two. By any of the approach you can say that the correct answer is magnetic moment by 2. Fourth option. Alright. Let's move on to the next. Yeah, that was the most calculative. I would say it was given in the problem, it was given in the paper in section B so that students can choose it to leave. So that the students can feel that, this is a problem which was meant to be left in the exam. So most calculative. But yeah, calculation is possible, obviously. We will see how. The interesting part is they are giving you the rated value, rated, effective, virtual value or RMS value. Rated, effective, virtual or RMS means the same. And what they are asking you is the peak current. So can I say here I naught they are asking? That should be E naught by XC. The E naught can be written as root 2 times ERMS and XC can be written as 1 by omega C. 1 by omega C. So the final answer will be root 2 ERMS. Omega, denominator will also become numerator, omega, which can be written as 2 pi f, and the C denominator will also be here. Now you are supposed to just keep the values intact. Root 2, ERMS you can write 210 into 2, pi can be written as 22 by 7, 7 will cancel 210 at 30 times, frequency is given as 50 and the capacitance value is 10 into 10 to the power minus 6. So you can see few things can easily cancel each other. Root 2, 1.414 you can approximate and then approximately your answer will match to 0.93. Okay, partial calculation ke liye I already gave you the hint. Remaining you can do in your free time, but as far as examination is concerned, this question you should be leaving here. Okay, that's an art to choose which are to be left. Okay, right, let's talk about the next. 2 heaters and it's a conventional problem here. It's a proper conventional problem. If you have prepared for the NEET, if you have done the NEET PYQs, then definitely you will be knowing those formula which I am going to use now. 2 heaters A and B have power rating of 1 kilowatt and 2 kilowatts respectively. Those two are first connected in series. So if you see P series, that will be given by P1 P2 by P1 plus P2. P1 P2 by P1 plus P2. So that's 2 into 1 by 2 plus 1. So that is 2 by 3 watts. Okay, and then they are connected in parallel. P parallel for the same voltage is given by P1 plus P2. So that will be 2 plus 1 and hence that is 3 watt. So what they are asking you is the ratio of P series by P parallel. So that is 2 by 3 by 3. Hence the answer is going to be 2 by 9. 2 by 9, unitless, dimensionless. It's the ratio. So 2:9. Fourth option is correct. Is that fine? All right. Chaliye. Move on to the next one. The VT graph, very simple question from motion in a straight line, or you can say the kinematics graph, which you people have worried about the entire year. But the question was very easy. So here in this portion, there is no acceleration. Here for this portion, I would say slope. Acceleration will be given by dv by dt. So that can be written as slope of VT curve. Okay, slope of VT curve. So you will say for this portion, acceleration is going to be a positive constant. For this portion, middle portion, acceleration is 0 because the slope is 0. and for this portion acceleration is going to be a negative constant. So you have to choose 0 positive constant, 0 negative constant. So 0 positive constant, 0 negative constant. The only option you had is option 1. Is it fine? All right, let's move on to the next one. If the mass of the bob in a simple pendulum is increased to thrice its original mass, I'll write the time period 2 pi under root L by G, independent of mass. Independent of mass. So whatever you are writing, if the mass of the simple pendulum, this from here to here, it's useless. Useless to change the mass. Okay. And its length is made half of its original length. This is something interesting. T is directly proportional to root L. So if length is made half times, this implies T final will be made 1 by root 2 times. Or that can be written as root 2 by, what they are asking, x by 2 times, half you want to do. Or you can write it as root 2 by 2 or 1 by 2 of root 2. See, basically you will say length is made half, since it is directly proportional to square root of length, T final will be 1 by root 2 times. But if you notice, they are saying x by 2 times. X by 2 times means you have to see that if you isolate half, what is the value of x? If you isolate half, what is the value of x? You had root 2, you wanted to isolate half, definitely you are supposed to multiply and divide by root 2. So what you got is half, which you can isolate, and hence by comparison, the value of x is root 2 inside the bracket. So the value of x is root 2. Is it fine? Hope you are able to understand the discussion clearly. Alright. Very soon we are going to complete that. Very interesting problem from the gravitation. Interesting के साथ I would say important भी और confusion भी है. Confusion attached to this problem is that which type of energy you need to consider. So if you have not gone through the problem, let me explain it to you. You are having and many other things in language are also important. For example, they are not saying a radius of r, they are saying an altitude of 2r. So altitude means height. So if you want to launch a satellite from the surface of Earth to a height equals to 2r, so you need to understand that the radius of this orbit effectively is going to be 3r. Okay. So you want to launch from here, and if you want the satellite to go from here to here in the orbit, definitely its total energy has been changed. Okay. So initially it was at rest. Now you want to give it some energy so that it will reach here. So can I say here energy required, delta E required will be equal to total energy finally minus total energy initially. Total energy finally is G capital M small m GMM by 2r minus of GMM by 2r, 2 into 3r. Because when you write GMM by 2r, there r is the radius of the orbit, and here effective radius of the orbit has become 3 capital R. Minus initially when you write the potential energy minus GMM by r, and when you write the kinetic energy at the surface is basically 0 because it was placed or it was about to get launched. Surface pe it is 0. So if you want to simplify that, that is minus of G capital M small m by 6r, minus will be plus GMM by r. So see, it is like 1 minus 1 by 6. 1 minus 1 by 6 is 5 by 6. 1 minus 1 by 6 is 5 by 6. Okay. Understood the tricky part in the question, that altitude means height of 2r. So basically the effective value of the radius is going to be 3r. It's a bit controversial problem. People are saying this to be this is a controversial statement, controversial situation. Okay, why it is controversial, this, that, we will talk about it later. Non-polar, non-conducting, like that. Here, let's try to identify let us choose the most appropriate. Okay, let us choose the most appropriate solutions, statements. Okay, so there is a sheet which is placed on the horizontal surface in front of a strong magnetic pole. So that's a sheet. Okay, and suppose that's a very strong magnetic pole, north pole, very strong. very strong. You have to consider this. Okay. A force is needed to hold the sheet if it is the magnetic. So if it's a magnetic sheet, definitely it will try to get attracted. It will try to get attracted and come nearby. But if you want to hold it there, definitely you have to apply some force. So yes, there is a force required. Hold the sheet there if it is non-magnetic. So if it is non-magnetic, even if you leave it, it's not gonna get attracted. So there is no external force which is required to make it stay there. So not required. Not required. Okay. Now, move the sheet away from the pole with uniform velocity if it is conducting. So if the sheet is conducting and you are moving it away, so definitely there will be some EMF induced, and if the EMF is induced, then definitely the Lenz's law will come into the picture, which will try to oppose the change responsible for its production. So why there will be an EMF induced? Because you are trying to move it. So what the induced EMF will try to do? Oppose the motion. So if you still want to make the motion continue away from it or towards it, it doesn't matter. You are changing the flux, that matters. So if you are trying to move it, you need to apply some force. The energy consideration, the force applied in motional EMF, like that. So definitely it is required. And here, why I said that it is controversial, the reason is, even if it is non-polar, there will be slight polarity induced because of this type of motion. But as far as the NCERT statements are concerned, you consider the electromagnetic induction only in the case of conducting bodies, because the charge carriers will be freely movable, they will induce the polarity and all those things. Okay, so for a safe side, the most safe options are A and C only. A and C only. Again, the D option is a bit controversial, but it's not the correct time that we get into too much technical aspect. You can choose the safe options for you. Okay. Let's hope the NEET also gives you that only. And I think we are done with all the, we are done? I think we are done. Let's see if we are left with, yeah, we are left with the last message. Okay. So you can also help out your friends who are still getting some wrong key confusion and this, that. So you can forward the discussion and then they can see the discussion and get on to the, they can arrive to a common conclusion for this. And do share, subscribe this channel. Definitely we have completed one session and now we are excited to meet you all for the upcoming session. Okay, so let's hope. Thank you so much. All the best. Sleep well. And remember, if today was not that much your day and you couldn't reach your expectation, then God is definitely having a better plan for you. So don't get disappointed. Keep going in life. All right. All the best. Stay smiling. Good night.